Welcome, friends and colleagues. It's so wonderful to see so many of you. My name is Yixian Zheng. As director of Carnegie's Embryology Department, it is a great pleasure for me to welcome all of you to today's symposium in honor of our dear colleague, Joe Gall's retirement. In the midst of pandemic, I'm very touched by the enthusiasm and care that Joe's former associates have shown in organizing this Zoom-based symposium to celebrate Joe's contribution to science. The symposium is organized by Joe's former trainees, but in the process, many of Joe's scientific friends have reached out so they can participate in today's celebration. We want to thank Navi Marbe, our communications coordinator, for helping with all logistics of the Zoom symposium, and also our IT for supporting the Zoom behind the scenes. I want to thank the organizing committee, Suzanne Gerbi, Ginger Zakian, Jilong Liu, and Zara Niazami for putting this symposium together. The organization was challenging because the committee members live on 3 different continents. Luckily, we have Suzanne Gerbi. As Joe puts it, as long as you have Suzanne, you'll have a great event. We will have a great event because of her. So please check your email from today's meeting agenda as a PDF. We had some minor changes to ensure the smooth running of the talks. We decided to use the time in the beginning to coordinate speakers. So this took the time away from the welcome slideshow, but you will be able to see that we'll host that slideshow on our private site. The event is fairly long, so the organizers have scheduled a few breaks. Our department has gotten to know what many of Joe's former trainees throughout the years, particularly by attending the past great symposia organized by the trainees for Joe's birthdays. Joe's academic family has become part of the extended academic family of our department and our institution. We have learned a lot about Joe's amazing legacies in science discoveries and in training generations of scientists. Many of Joe's trainees have themselves been contributing to science throughout the world. For such a long and illustrious career, Joe has made so many contributions to science. So each time when a symposium like this is held, I find myself to learn something that Joe has discovered that I actually missed. So I know today is no exception. Although many of us had not had the fortune to train and study with Joe, we treasure having Joe as our colleague. I have learned a lot from Joe in the last 24 years at Carnegie. Each of us have our own memories about Joe. For me, Joe had played an important mentoring role in helping me to cope with stress as a beginning faculty. He has always encouraged me to explore the biological questions that are important but understudied. We're lucky that in retirement, Joe will continue to do research. So we'll continue to enjoy discussing science and life with Joe at lunch and at our Friday gatherings once our life returns to normal. We are looking forward to interacting with Joe's former trainees as part of our extended family in years to come. Now I want to introduce our president, Dr. Eric Isaacs, who will give some remarks. Eric joined our institution over 2 years ago. He is a physicist and had directed research at the famous Bell Labs before joining the Argonne National Lab. Prior to coming to Carnegie, Eric was provost and vice president for research at University of Chicago. We are very happy that Eric is able to find time in his very busy schedule to help us kickstart this symposium. Here's Eric.
Thank you so much, Yixian. And I do want to say it's a real honor for me today as we gather to honor our colleague, Joseph Gall, on his retirement. And I want to begin by offering my sincere congratulations and best wishes to both Joe and to Diane. And I believe I speak for everybody here at Carnegie in saying how much we admire you, Joe, and how much you have inspired all of our science for many years. And you just heard an example of that from Yixian. And I think the inspiration has come from both your work, fantastic work, but also through your character. And so I'd like to just say a few words. I did a little research here. Say a few words about why I'm so honored to be here. And I want to start with something that I read in Journal of Cell Science, which was celebrating Joe's 75th birthday. I believe it was written by two of his former students. And they made a very simple statement, which is that they called him a scientist. And I can't think, as a scientist myself, of a higher praise for somebody. So it's at events like this that I know it's appropriate to offer some flattering assessments of the guest of honor and their work, and maybe even polite exaggeration for their achievements. But it is really simply impossible in this case to overstate the impact Joe has had in his life sciences. There's no reason to be polite. It's not, nor exaggerate. When he won the Lasker Award in 2006, he was called the founder of modern biology. And I thought about that for a while. It's really remarkable because, Joe, when you were a student, I believe biologists were just starting to ask basic questions about DNA, RNA, proteins, how they worked, what they were doing. And a lot of that work focused on single cells. And so Joe, in those days, took a microscope that he had built himself, and he started looking at cell nuclei. And as you all know, the rest is history. He became one of the world leaders in understanding, through careful, methodical, and brilliantly transforming our understanding of the mechanics of life itself, particularly starting with the nucleus. His contributions to microscopy have been remarkable. I'm a physicist and I've done microscopy, and what he's done have made him a luminary in this field throughout his career. And he's made this he has an extraordinary gift for asking not just how to build stuff, but also really important questions. And through that has identified many appropriate organisms, and particularly something that embryology is known for, model systems that elucidate some of the key questions that he has asked. And so in developing novel and ingenious tools and asking key questions, he's really had a remarkable impact on the science itself. But there's something I really want to emphasize, which I really, I'm totally in awe of in Joe's ability, not just to transform biology through his exceptional acumen for the science and instrumentation, but really through his teaching and mentorship, and especially his teaching of and mentorship of women scientists. In one interview that I read, Joe had credited his mother for instilling his lifelong belief that women could and should be great scientists. So when he became an adult, he saw nothing unusual in a woman excelling in a science career. And in this way, Joe opened the doors of his lab, to any brilliant researchers who wanted to join him, whether male or female. And he treated all of his students with respect and set the highest standards and did an amazing job nurturing their talents. And the results speak for themselves, and you're going to hear from some of those people today. Generations of women scientists have credited Joe for training them, for working with them, for mentoring them, for supporting them, giving them advice, encouragement. And as I said, you'll hear from some of those people today. Some of them, like the three women students, Mary Lou Pardue, Susan Gerbi, and Elizabeth Blackburn, have all gone on to serve as president of the American Society for Cell Biology. Mary Lou Pardue has noted that the year she was elected to the National Academy of Sciences, there were 3 women in her class. 2 of them worked with Joe. Really remarkable. And of course, his former postdoc, who's here with us today, Elizabeth Blackburn, shared the Nobel Prize in Medicine in 2009, and we'll hear more from her later on. In recognition of this work in his superlative record of training and mentoring women in sciences, Joe also received the AAAS Lifetime Mentor Award and the ASCB Women in Cell Biology Lifetime Achievement Award, and he is a member of the Rosalind Franklin Society. And I think that whose mission it is, as you know, to promote the contributions of women in the life sciences. So you can go on and on. He's had an amazing career. And I think what really stands out is not just his own science, but what he's given to the people who he's mentored, who he's trained, and who he's worked with through his career. In case no one else says it, and not to embarrass you too much, Joe, I do want to mention some of the other achievements aside from the Lasker Foundation Special Achievement Award. Also, the Society for Developmental Biology Lifetime Achievement Award, the E.B. Wilson Medal from the American Society for Cell Biology, the Wilbur Cross Medal from Yale, and then he shared the 2007 Louisa Gross Horowitz Prize from Columbia, and is a member of the AAAS, the National Academy, and the American Philosophical Society. So, you know, in ending, I'd just say that, and you heard this from Yixian, what Joe has brought to Carnegie, where he's been since 1983, is just, you can't evaluate it. It's just so great. He's just highly valued by his colleagues here in the department and even across the whole institution who know of his work, who know that he's been such a leader. And, you know, part of my job in the first 2 years is to figure out what makes Carnegie different, what makes Carnegie really special. And I've talked to a lot of our scientists, and of course, everyone says, well, you know it when you see it. But I think when you look at someone like Joe, it is so clear that he instantiates what it is to have, to be a Carnegie scientist. His brilliance, his independence, his innovative approach to really to research, his generosity with his colleagues, his willingness to sit and talk and debate, and lastly, his steadfast commitment to the highest principles of scientific integrity. So I'd like to say, Joe, that as president of this great institution, it is really my honor and my privilege to thank you for the many years of exceptional service. I want to wish you and Diane a healthy, happy, and well-earned retirement, but I also know that you're going to continue some of your research in the coming year. So again, thank you very much.
So I now want to introduce Dan Brown. Before that, I want to thank Eric for helping us to do this, to kick this event off. Our next speaker is Dr. Dan Brown. Dan really needs no introduction to us biologists. He created the legacy of our department by defining a new research frontier and by hiring many of the smartest scientists in the nation. These include Alan Spratling, who is still in our department, Steve McKnight, Doug Koshland, and Andy Fire. Importantly, Dan also hired Joe. Dan will make some remarks and introduce Joe. Here's Dan.
Joe Gall was a professor at Yale when he came to our department on sabbatical in the 1980s. When his year at our department was up, he returned to Yale, but he was in line to be chairman of biology, and he rebelled against that idea. I got wind of his dilemma and offered him a staff position. Without a doubt, this is one of the best things that I did as chairman of the Department of Embryology. It meant reducing the size of his lab and the number of students, but it also meant that he had no administrative duties here. He wanted to stay in the lab and certainly not become a chairman. I am proud of my decision to hire Joe. He may be retiring at the age of 92, but he's still working in the lab. He remains the expert on anything optical, and that includes the latest innovations. In fact, Joe has given the only course in our department, and it has been taken by all of the graduate and postdoctoral fellows. Joe will continue working at the bench, doing important research. Congratulations, Joe, on a wonderful and continuing career.
First, I'd like to thank Yixian, Don, and Eric for their comments and for their support over the years. Second, I'd add my thanks to the organizers of this meeting, Susan, Ginger, Jilong, and Zara, and to Naveed for his technical support. It's great to see, that is, with quotes, so many colleagues, students, and friends, at least virtually. And thanks for attending. I'm pleased to give a short presentation now, a summary of my life and just some of the work in our lab, work to which you have all contributed. So let's begin. I call this presentation Through the Looking Glass, which many of you will know is the title of the 19th century book by Lewis Carroll that describes the adventures of Alice, who enters through a looking glass, what we would now call a mirror, into a strange and wonderful world of mythical creatures and events. In much the same way, the microscope for me, and for many others, has been the way we have entered the wonderful world of microscopic objects and phenomena. Here's a photograph of me taken when I was about 14, looking through my real microscope, not a toy one. This was the middle of the Second World War, and I was fortunate not only to be too young to be drafted, but to have resourceful parents who were able to find and purchase this instrument for me. I had spent many hours looking through the microscope, preparing specimens for observation, reading about the things that I was observing, and becoming a scientist on my own. I think one of the greatest things about this experience was the independence that it fostered. If I wanted to know something, I had to either find it out myself or read about it in one of the books my parents were able to provide for me. Skipping ahead a number of years to the time I entered graduate school in the old zoology department at Yale, my advisor, Donald Polson, was a Drosophila geneticist, and for a long time I thought I would do a thesis in genetics. But somehow I came across a textbook that contained this image of a chromosome from a salamander oocyte. It was made by an investigator by the name of William Dury, and it showed a chromosome from an oocyte of a salamander being stretched between microneedles. I thought surely the scale marker was wrong, that the chromosome couldn't be this large. But this set me on a literature search, and I found that indeed giant chromosomes had been observed in the oocytes of frogs and salamanders as far back as the late 19th century. They had been given the quaint name of Lampbrush chromosomes because of their imagined resemblance to the brushes used at that time for cleaning the soot from oil lamps. This has remained the name for these remarkable structures. Giant Lampbrush chromosomes have been found in the oocytes of frogs and salamanders, and indeed in a variety of other organisms that have giant germ cells. Although not so well known as those from amphibians, Lampbrush chromosomes are found in many birds, fishes, and even in a few plant species. Most of my work has been done on the two species shown in this slide, the frog Xenopus and the salamander Ambystoma. Here we see a bit of ovary from the frog Xenopus, showing oocytes of all different sizes, from the very small transparent ones to the largest yolk-filled ones, which reach a size of 1 millimeter or so in diameter. And for those of you not familiar with animal reproduction, I remind you that despite their size, these are single cells with a single nucleus. This slide shows how easy it is to remove the giant nucleus by poking a hole in the oocyte and squeezing. The nucleus pops out, often completely free of yolk, as in this example. This nucleus is almost always called the germinal vesicle, or GV, a term that actually goes all the way back to the early 19th century. It was first described from oocytes of the chicken, long before it was recognized as the nucleus, and even before the oocyte itself was recognized as a single giant cell. This slide shows the contents of a single GV of the frog Xenopus. At this stage of early meiosis, the maternal and paternal chromosomes are paired. That is, each nucleus contains the haploid number of paired chromosomes. I point out the numerous small bodies in the background, as we will come back to these a bit later. Here is a higher magnification image of a portion of one chromosome pair from the salamander Notophthalmus. Notophthalmus is also known as the newt and is a fairly common inhabitant of ponds and streams throughout eastern North America. Here you can see loops of chromatin extending laterally from the axis of the chromosome. Each of these loops represents a single active gene, visible because it is covered with the ribonucleoprotein products of transcription. Here's a diagram elaborate chromosome structure. At the right is shown a single pair of loops, which represents an active gene covered with the products of transcription. Transcription begins at the thin end of the loop and continues all the way around the loop to the thick end. It should be noted that despite the size and prominence of the transcription unit, transcription loops, most of the DNA of the chromosome is tightly wound up and is transcriptionally inactive. In addition to the Lampbrush chromosomes, each oocyte nucleus contains hundreds to thousands of small bodies of different types. The largest of these have been called nucleoli since the 19th century, although the relationship to the nucleolus of ordinary somatic cells was not clear at all. To illustrate the extreme difference between the oocyte and a typical somatic cell, I show you an image taken from a classic paper by Barbara McClintock of the nucleolus of maize, or corn if you prefer that name. Here, there is a single nucleolus attached to a specific point on one chromosome, the so-called nucleolus organizer region. What then is the relationship between the single nucleolus shown here and the hundreds of nucleoli in the amphibian oocyte? The answer to that question takes us to the earliest stages of the oocyte, long before the giant Lampbrush chromosomes are so prominent. At this time, we can see the chromosomes as thin fibers. On one side of the nucleus, however, is a large mass of chromatin, that is, DNA plus protein. To make a long story short, what has happened here is that somehow the genes coding for ribosomal RNA have gotten out of the chromosome and replicated independently. They have formed a large mass on one side of the nucleus, consisting of thousands of copies of the genes coding for ribosomal RNA. This mass of DNA is referred to as amplified rDNA, or amplified ribosomal DNA. Here's another view of early oocytes from Xenopus, showing the stages of ribosomal DNA amplification. In this case, the ribosomal DNA has been labeled with a radioactive tracer, shown here as black dots in the photographic emulsion that records the radioactive molecules. This technique of autoradiography would have been familiar to an earlier generation of cell biologists, but is much less commonly used nowadays. Another way to look at the amplified ribosomal genes is by electron microscopy. This image is a quite famous one produced by Oscar Miller and reproduced in numerous cell biology texts. It shows individual ribosomal RNA genes undergoing active transcription. The active transcriptions get longer and longer as they move down the gene, giving rise to these iconic Christmas trees. Most of the early studies on specific genes were carried out on the genes coding for ribosomal RNA. This was because these genes were present in hundreds to thousands of copies per nucleus. One other type of DNA was available for molecular analysis in those early days. This is the DNA known as satellite DNA, and this image of satellite DNA would have been familiar to an earlier generation of cell biologists whose only way to separate different fractions of DNA was by centrifugation. It turns out that about 10% of the DNA in the mouse genome has a different density from the rest of the DNA and can be separated by centrifugation, shown here as a satellite peak. When this satellite DNA was purified and used as a probe on cytological preparation, it was found to hybridize to the ends of the mouse chromosomes. Satellite DNA was found to consist of very simple sequences and essentially defines large regions of gene-free chromatin. It turned out that chromosomes of many organisms had regions of satellite DNA. Here is another well-known example from the fly, Drosophila virilis. Roughly half of each chromosome consists of simple sequence DNA that does not contain genes. Those are the large, dark regions occupying about half of each of the chromosomes. I want to make a digression here to look at the single-celled organism Tetrahymena. For a period of about 10 years, starting in the early 1980s, I turned my attention to Tetrahymena. It had been known for many years that Tetrahymena has two nuclei, a small genetic nucleus or micronucleus, and a larger macronucleus that contains a massive amount of DNA. I was struck by the fact that the macronucleus had multiple nucleoli and in fact looked very much like the nucleus of an oocyte. To make a long story short, it turned out that the macronucleus of Tetrahymena contains many thousands of copies of the genes coding for ribosomal RNA. These are present in multiple nucleoli on the periphery of the macronucleus, a situation very similar to that in the amphibian oocyte. When we isolated the ribosomal genes of Tetrahymena, we found that they were in pairs located head to head. The tails turned out to be quite interesting. This slide shows the sequence of the Tetrahymena telomeres. They contain many copies of a repeated sequence as shown by Elizabeth Blackburn, at that time a postdoc in my lab. That sequence was C4A2 on one strand and G4T2 on the opposite. At that time, we did not know it, but later Liz Blackburn and her student Carol Greider found that this and similar sequences characterize the ends of chromosomes or telomeres in many other organisms. As many of you know, this finding led eventually to the Nobel Prize for Liz, Carol, and Jack Shostak. I want to come back to the germinal vesicle and the organelles that it contains. We already talked about the many free nucleoli that occur in the germinal vesicle. There are two other major bodies in the germinal vesicle, which we call the histone locus body and the speckle. The histone locus bodies get their name from the fact that one or two are attached to the Lampbrush chromosomes at a specific locus, in addition to the several hundred copies found free in the nucleoplasm. Here's a histone locus body with attached speckles, shown by differential interference microscopy in the upper left image, and after specific staining in the other three panels. The histone locus body contains the well-known protein named coilin, as well as SM proteins associated with small nuclear RNAs. I want to close this presentation by briefly mentioning molecular studies on isolated germinal vesicles. As I showed earlier, we used isolated GVs for cytological studies of the chromosomes and nuclear organelles. We've also studied isolated GVs by molecular techniques, which I can only touch upon here today. This slide shows the isolation of the GV from the Xenopus oocyte and removal of the nuclear envelope to give a fraction of pure nuclear RNA. One of the surprising findings from studies of isolated GVs is the occurrence of stable intronic sequences derived from splicing of many genes. These intronic sequences are found not only in the nucleus where they are formed, but in the cytoplasm. At the moment, we have no good hypothesis for the occurrence of these sequences. I wanted to close on something we know very little about to emphasize that the oocyte contains many secrets and that we can expect many surprises as we continue to oocyte its wonderful chromosomes and the many molecular processes occurring in it. And that ends my slide presentation.
Well, thanks, Joe. It's always amazing to hear your reflection on your past research experience. Joe has been really humble throughout his life, and he mentioned that the in situ hybridization is less popular now at the earlier part of his talk. I want to say, actually, that's not true. In situ, the concept and basic methodology of in situ hybridization is just as popular now as before, because everybody is using this based on this methodology to develop ways to visualize genome in 3D, which really has been very burgeoning in the field. So now I want to introduce Ginger Yixian. She is one of the organizing committee members. Ginger will introduce and moderate the reflections and anecdotes given by a number of Joe's former trainees from different era. Ginger studied with Joe as a graduate student at Yale from 1966 to 1970. After postdoctoral training, Ginger joined the faculty at Fred Hutchinson Cancer Research Center. She later joined Princeton University and became Harry Weiss Professor of Life Sciences at Princeton. Ginger is a great example of Joe's former trainees who have made important contributions to science. Ginger's research is critical to our understanding of telomeres. She was awarded the Women in Cell Biology Senior Award and was elected to National Academy of Sciences. She has served on many leadership roles, including ASCP Council, and also the Board of Directors of Genetic Society of America. Here's Ginger.
Okay, thank you, Yixian, for that introduction. And to all of those of you who have made this meeting possible. And finally, thank you to Joe for accepting me as a graduate student in his lab almost 50 years ago. I hope you and Diane have a wonderful retirement. Now, as has already been mentioned, Joe won the 1996 AAAS Lifetime Achievement Award in Mentoring. And the following speakers that I'll be introducing are all former Gall lab members, and their accomplishments provide a wonderful example of Joe's mentoring abilities. Our first speaker is Joan Steitz, who will talk about the early years at the University of Minnesota. Joan's family lived in Minneapolis, and she spent the summer after college graduation doing research in Joe's lab. She then received her PhD from Harvard, where she was the first female graduate student in Jim Watson's lab, and then did a postdoc with Francis Crick at the MRC. She joined the Yale faculty in 1970, where she is in HHMI-supported research. Her research focuses on RNA, where she has made multiple pioneering findings. Among these was the technical tour de force demonstration that the initiation of bacterial protein synthesis involves base pairing between the 16S ribosomal RNA and mRNA. Joan went on to discover and characterize the small nuclear RNAs involved in splicing. Her large number of awards include membership in the National Academy of Sciences and the British Royal Society, and receipt of the National Medal of Science. Joan.
Unmute and start video. So am I visible to people now?
Yep, we can see you and hear you.
Okay. So I guess I want to move on quite quickly to sharing my screen. So I will do that. Well, let me first say, I mean, this is such a magnificent day. And it's been organized so very well. And thank you, Ginger, for the lovely introduction. And thank you to all the rest of you, especially Joe, for highlighting just some of his accomplishments. He absolutely could not do it all in a short period of time. And we only heard about some of them. But let me now go on to share my screen here. see. Okay. We do this. Okay. That should work to get it presentation mode. There we go. Okay. So let me just start out by saying I feel so lucky to have known Joe. I'll be telling you why. And I want to say congratulations to him on this very special occasion. And I have always considered Joe to be an absolutely inspirational scientist, not just based on the importance of the problems that he's decided to work on, but also about the way in which he does science and treats his colleagues. And I'll have more to say about that later. As you heard, I only spent 2 short months in the summer of 1963 at the University of Minnesota in Joe Gall's lab. I had just graduated from Antioch College and was slated to begin Harvard Medical School in the fall. But I wanted to spend a couple of months at home with my parents. I came from Minneapolis. And so I talked to several people at the University of Minnesota about the possibility of working in their lab. Meanwhile, while at Antioch, I had been introduced to the very exciting new field of molecular biology through Antioch's co-op work-study program, where students were sent off into the real world to gain experience, not just studying on campus. And I had been very fortunate to have been assigned a lab job with Alex Rich at MIT, which is where I first heard about double-stranded DNA and all the things that it might mean for genetics. So I had interviewed several people at the University of Minnesota. Joe was the only one who said yes. And so I went to his lab in late June. What I knew about Joe at that time was that he was a rising young leader. He was about to move from Minnesota to Yale. And he just published this wonderful experiment diagnosing the kinetics of degradation of DNA by DNase, which had told him and us that chromosomal DNA is in fact a single double-stranded molecule. What I didn't know about Joe was that that summer in his lab would change the entire course of my life. So what Joe did was to give me my own project. This was to isolate ciliary basal bodies from Tetrahymena piriformis, and you've already heard Joe mention Tetrahymena, and look at the preparations for their DNA and RNA content. The reason that that was salient at that time was because DNA had just been discovered in mitochondria, and the question was how many other subcellular organelles would likewise contain important DNA. There had actually already been a paper published on this subject by Seaman in experimental cell research, where he had done exactly the same thing, but Joe was doubtful that the purity of Seaman's preparations of ciliary basal bodies were great enough to give the right answer. They had shown a little bit of DNA and a little bit of RNA. And what I was supposed to do was do basically the same things, look at the steps carefully, and particularly at the end, look in the EM at the apparent purity of the preparation. Joe spent most of the summer packing up his stuff and moving it to Yale. So I was left pretty much under the guidance of a single postdoc in the lab, Steve Wolf. By August 1st, it turns out that I was so enthralled with doing my own experiments and discovering things in science, I decided that I did not want to go to medical school. I wanted to get a PhD instead and do science. And it was Joe who largely orchestrated my switch from Harvard Medical School into a new program at Harvard in biochemistry and molecular biology for the PhD. At the times I did see Joe, I was always impressed by the, as you all know, by the breadth of his knowledge and interest in all aspects of science. One thing, for instance, that he knew was that during a partial eclipse of the sun, the shadows would come out in these beautiful crescent shape. And so he hauled me outside of the zoology building and said, I must take your picture. And here it is. Really a remarkable picture. Thank you, Joe. I've treasured it my entire life. Another thing Joe liked to do was sort of set thought problems for people. And I remember one of the thought problems he set for me was, if you were caught in a rainstorm, would you stay drier by walking at a normal rate or by running, which would mean you would encounter all these raindrops coming at you horizontally as well as the vertical ones going down on your head? And my knowledge of differential equations was not as good as Joe's, so I could not solve the problem mathematically. But I think the answer is that if you get out of the rain fast, you stay drier, even though you might have encountered a lot of horizontal drops. Let's see. So I went off to graduate school in the fall of 1963. And Joe decided that the results I had on the purification of the basal bodies and their DNA and RNA contents should be published. And in fact, that work did appear in very early 1965 in the Journal of Cell Biology. And here is the, the sort of title of the article. You will notice that Joe's name does not appear on this, nor did it even appear in the acknowledgements. Here's some pictures down below of the isolated basal bodies, and on the right, the EM image of what the EM looked like. They were higher, more highly purified than the previous publication, and they had less DNA and RNA in them. And as you know, it took then decades for the cell biological community to decide that probably basal bodies and their paralog centrioles do not contain nucleic acid. But in the process, I had written, of course, a draft of the paper. Joe had completely rewritten it. It's totally awful. And despite that fact, as I said, his name appears nowhere on the paper. But this is Joe Gall. So I'd like to just end with a wonderful picture, and you've actually already heard this picture alluded to, of Joe Gall, me, Mary Lou Pardue, and Oscar Miller. And I'm pretty sure that this was at the I know it was at the National Academy of Sciences. I think it was probably 1983. And so here you see two of the three women that were inducted into the National Academy that year. So I'd just like to end by saying to Joe how thankful I am for a life in science and for continuing to inspire me, and as I know he will in the future as well. Thank you very much, Joe.
Thank you, Joan, for that wonderful presentation. Our next speaker is Liz Rogers, who will talk about the transition from Minnesota to Yale. After her undergraduate years in zoology at Oxford, Liz came to Joe's lab in Minnesota in 1962 for her PhD, and then moved with Joe to Yale in 1963. Her graduate research focused on ribonuclear protein particles in the nucleoli of amphibian oocytes. She continued to study RNPs, first as a postdoc and then later as a faculty member at the University of Edinburgh. Far ahead of her time, she also developed and ran a 13-year program that was a collaboration between the university and the city primary schools on environmental issues. Later, she reinvented herself as a primate biologist. For 20 years, she studied great apes in Central Africa. Liz.
Hi, everyone. Can you hear me?
Yes.
Okay. It's funny not having an audience in front of me, but hello. And well, I'm really pleased to be joining you today from the UK, where we incidentally have an enormous thunderstorm going on. So I'm talking about the change from the University of Minnesota to Yale from 1962, when I first arrived as a just graduated zoology student from Oxford in the UK. So you may wonder, what was I doing working with Joe? Well, it was because I had heard a lecture from Mick Callan, who was then professor of zoology at St Andrews University in Scotland, about the Lampbrush chromosomes when I was a student. And I was completely blown away by these things. And I decided that I wanted to travel, but I also wanted to find out more about them. I had not liked the genetics lectures at Oxford when I was a student. They were very classical and dry. And believe it or not, in 1959, 60, something like that, DNA was hardly mentioned. So to see these chromosomes actually at work, as it were, was to me absolutely amazing. Now, to give this talk, this is going to be perhaps a bit different from other people's talks. I did not stay in the field, as you've heard, so I had to find a source of information more or less from the time. And I knew I had a treasure trove in the form of my letters to my parents, that from fall 1962, I wrote to my parents every week describing what I was doing. And so you have to think of it through that sort of filter, but this is what I have derived, the sense of what it was like being in Joe's lab at Minnesota and then Yale. I've derived that from my letters, but there's not as much scientific detail as you will see. But at least they give this sense of what it was being like, what it was like in being in Joe's lab. From day one, I say to my parents how enjoyable it was talking to him about his work because of his enthusiasm and his clarity and his broad interests. As a student at Oxford, I had studied the whole animal kingdom, which you could do in those days. And I had been interested in a large proportion of the different animals in it. So I was delighted to find that Joe worked on a range of animals, including not just amphibia, as you can see, these newts here, giving the sense of the target animal we were using then. But not just amphibians, but snails, cycads, plants, for goodness sake, insects, ferns, and he chose the species according to which cellular puzzle he decided to investigate. For example, as Joan has just said, the origin of centrioles and basal bodies, for which he needed cells with many cilia or flagellae. And so you had exotic spermatozoa and protozoa like tetrahymena all being worked on. So let's progress to the next slide. And I scanned this picture from Joe's 1954 paper because I wanted you to see on the top there how beautiful the germinal vesicle, the isolated germinal vesicle is. And it was, as I say, completely mind-blowing to me that you could dissect this thing out by hand. Joe was at the time interested in the nuclear membrane as well as the nucleoli. And in fact, Mick Callan was visiting his lab in fall 1962 as well. First of all, I had to learn how to acquaint myself with the inverted phase microscope. And Joe went about this by giving me a completely disarticulated one, which I had to put together myself. And he thought this would do me good, which it did. I write to my parents in absolute joy and wonder about the first time I managed to make a preparation of Lampbrush chromosomes. And so I start with talking about getting the nucleus out, and then I say, if you do the tearing of the nucleus properly, you see the chromosomes unfolding and everything moves. The loops move in and out as you watch. I could have watched those things for an age. In fact, I did sit and watch them for at least half an hour. I thought, what are they doing and why? And Joe was so enthusiastic about this whole thing, and he had invented the techniques, and it was just extraordinary to be working there with him. one big question that was circulating at the time was, what was all the protein on the Lampbrush chromosomes doing? There was also great interest throughout cell biology in the possible regulation of gene activity by proteins, particularly the inhibition of it. And then there were all the questions about ribosomes, were they assembled in the nucleus or the cytoplasm? And the extra nucleoli in the oocyte nucleus were obviously a key to the answer. Now, I know Joe showed this image as well, but I had to put it in because in memory of Oscar Miller, who sadly has passed away, his iconic electron microphotographs of the ribonucleic acid, the ribosomal RNA being made, I've never forgotten. Of course, they came later, but I put it in honor of both Oscar and Joe, because Oscar had worked in Joe's lab in the summer of 1962, just before I came. In fall 1962, as I say, Mick Callan was visiting. We were doing experiments with isotopic labeling studies with C14 amino acids and tritiated uridine for RNA, asking where did all the radioactive precursors end up. Joe was visiting Yale and announced he was going to move. And so that put me in a quandary because, you know, was it possible for me to go with him? And eventually in January 63, my position, future position at Yale as a teaching assistant in Joe's cytology course was confirmed. By the time we were free of semesters and teaching students and things, we had exciting results of isotopic experiments in June, July 1963, and Mick Callan was getting similar results. Unfortunately, my letters to my parents don't really give any details, so I'm not quite sure what we found. But anyway, it was a good summer, that June and July, doing research. As Joan said, in August 1963, we packed up everything and moved to New Haven. And my sister came out and we drove in a VW Beetle via Vancouver Island to New Haven, as one does. Here we are in Yale. I found this cutting in a box of papers from the 1960s, which announces the appointment of Joe as professor of biology at Yale. And I love on the cutting, if you look at the bottom right half of it, I love this, Professor Gall will teach both graduates and undergraduates. And he added, I hope to get a fair amount of research done, which of course is what he did. Joe's cytology course was very popular. And there was a splendid occasion when a lot of comb jellies or tenaphores arrived in the lab. We hadn't actually ordered them. We'd simply asked if they were available, but there they were. And so I found myself with an aquarium full of comb jellies with their cilia beating away most beautifully. And Joe was interested whether if you cut off a comb, would it regenerate? So we found ourselves looking at comb jelly cilia under the microscope. My research continued where we'd left off. I swapped newts for axolotls for sundry practical reasons. And then in February 1964, my first mention in my letters to my parents about isolating the particles responsible for protein synthesis, which of course turned out to be ribosomal precursors. And that was analyzing those particles was my thesis project. And then by the time I'd passed comprehensives and the language reading science in French and German business, then by October the 64, I could express joy at being a full-blown researcher. I may say, as an indication of Joe's wonderfully broad interests, that the exams we had to pass for reading scientific German in other languages, he came into that as well because of his enormous collection of antique books on 19th century cytology, which we were able to translate for the exam. And so at Yale, by the fall of 1964, we were in full flight, and along came a lot of other people interested in Joe's work, from whom you will now hear. Thanks, Joe, you were a fantastic supporter and a great supervisor, and many congratulations on your long and inspirational career. And may you and Diane have a wonderful retirement. Goodbye.
Penjin.
Thank you, Liz. Our next speaker is Susan Gerbi, who will talk about in situ hybridization with input from Mary Lou Pardue. Why does it say Geelong up there? Did we?
Fixed. Sorry, I just tested my microphone. It was a problem.
Oh, okay. But
sorry.
Everything's okay? Okay. And I want to agree with Yixian that that in situ hybridization in its current guise of fish has not been put to the wayside, because many labs all over the world still use it. And I was finally able to publish a paper using fish, much to my great satisfaction this year. Okay, so Susan came to Yale in 1965, the same year as her fellow graduate student and friend, Mary Lou Pardue. Susan and Mary Lou played key roles in Joe's development of in situ hybridization, one of the most impactful contributions of his career. Susan and Mary Lou both assumed faculty positions in 1972, Susan at Brown University and Mary Lou at MIT. As a bit of an aside, Joan Steitz, Susan and Mary Lou were among the first women to get tenure-track positions at major research institutions. And I speak from experience that they were important role models for the next wave of female students, both in Joe's lab and elsewhere. Susan has led her department and university in many ways, including as the founding chair of the Department of Molecular Biology, Cell Biology, and Biochemistry. Research in Susan's lab focuses on ribosomal RNA. Her lab was the first to sequence a metazoan 28S ribosomal RNA molecule, and on a variety of aspects of DNA replication. Susan has found time to be a national leader in biomedical graduate education, guiding FASEB in developing best practices for graduate education. Like Mary Lou and Liz Blackburn, Susan followed in Joe's footsteps by serving as president of the American Society for Cell Biology. As you've heard, although there are 4 organizers for this celebratory meeting, Susan was the driving force. Thank you, Susan. We look forward to your talk.
Well, thank you, Ginger, for the nice introduction, and thank you, Joe, for allowing us to have this wonderful, fun event. It's too bad we're not in person, but it's great that we can do it by a Zoom webinar. So, sorry, let me back up. So as you heard from Ginger, both Mary Lou, my classmate Mary Lou Pardue, who is unable to participate today, and I'll share recollections from her as well. And I arrived in Joe's lab at Yale in 1965, and we both left with our PhDs in 1970. I came to Joe's lab knowing that I wanted to study chromosomes, and I was particularly taken by his work with giant chromosomes, namely the Lampbrush chromosomes that you've already heard about from amphibian oocytes. In fact, I ended up not studying Lampbrush chromosomes, but they've continued to be a wonderful model, as you've heard from Joe and others. So you've already seen the picture from Joe of the schematic of a Lampbrush chromosome. And he used this as a model to answer the question, how many strands of DNA are in one chromatid? This experiment was done by following the kinetics of DNA's digestion of new Lampbrush chromosomes and was published in Nature in 1963. You've already heard the result of this from Joan, who alluded to it. And let me show you a picture from that work. So if you follow over time the digestion of the loop or the main axis by DNA, so you can derive the kinetics. And from that, Joe concluded by looking at the slope of the plot that it was a unanine model, that is 1 DNA helix per chromatid. And it's something that we all take for granted now, but it was actually Joe who discovered this. However, shortly after I arrived at Yale, there was a visit to the lab by John Heddle, who had posited and had some information that perhaps it wasn't a unanine model, it was a binding model, according to Heddle. And so Joe invited Heddle to Yale so they could repeat the experiment and seek out the scientific truth. And this is a wonderful example of how Joe is always driven by finding the scientific truth, and that's much more important to him than his own ego, even if he might have proven to be wrong. But in fact, he was right, as one might have expected, and he got the same result as he had previously published. And it turned out that the data is always right if done carefully, but it's the interpretation that leads one to be cautious in terms of whether you're interpreting the results correctly. And so the interpretation is what differed, because Joe drew his plot as a straight line, y equals mx plus b, where the slope were the number of DNA strands in a chromatid. Instead, John Heddle thought that DNA might lose activity over time, and so he drew his results as a curve, and doing that, he concluded that it was a binding model. But we now know that Joe was correct, it's a unanine model. Let me now turn to the development of in situ hybridization, and I was privileged to be in Joe's lab at this time. The field was ripe for the idea because biochemical molecular hybridization had just been developed by Gillespie and Spiegelman, where RNA would hybridize to single-stranded DNA. And so the question was, how could one adapt this to chromosomes? Joe went off to a meeting in South America, and there at one of the evening social events, he and some other scientists were sitting around talking about the problem and the possibilities that maybe something akin to this could be developed to look at hybridization in chromosomes. Everybody who had been talking about this in that group at the meeting ran home to their labs after the meeting to try and develop the method. And they got stuck at the very first step. They were trying to figure out if they could make the chromosomal DNA that is normally double-stranded, single-stranded by denaturation, and then how to assay that it was single-stranded. And they tried various things like acridine orange dye, which can have a different color if the DNA is single-stranded or double-stranded. Instead, Joe, being the biologist, said, let biology be your guide. And that was the key for success. He used a biological system where he knew the answer so he could then work out the details of the method. And it was already known from work by Ratosa and Spiegelman in flies and Wallace and Bernstein in Xenopus that ribosomal DNA was associated with the nucleolus organizer region. Moreover, it had been described by King in 1908 and then followed up later in 1942 by Painter and Taylor that, that there was extra chromatin in the meiotic packaging stage of the toad bufo oocytes. And you can see the extra nucleoli shown near the nuclear periphery in a Lampbrush chromosome, more in the interior. And Painter and Taylor said that each nucleolus showed a foil-gin positive or DNA granule that was directed towards the nucleoli at the nucleolar wall. So Joe, together with Mick Callan, tried to repeat this finding on salamander oocytes, but were unable to do so. However, Joe then got some decades-old dyes as a gift from Painter while I was a first-year student at Yale. And to his great delight, he saw foil-gin positive DNA in the new extrachromosomal nucleoli, suggesting that that might be ribosomal DNA. He was so excited, he ran around the lab to find others that he could share his observation with in the microscope. Nobody else was there except for me. I was getting ready to go to class and I was already late. But Joe insisted that I should be late for class and go and look under the microscope and see this phenomenal result of Feulgen staining within the extra chromosomal nucleoli. And it was worth being late for class to see that. Subsequently, Joe's former postdoctoral student, Oscar Miller, showed DNA in nucleoli by electron microscopy, as you've already seen from some of the previous speakers. So the first observation that Joe published on this was in 1968, and you've already seen these pictures from Joe, where on the left you see the Pachytene oocyte, and it had tritiated thymidine silver grains over the cap, which were the extra chromosomal nucleoli all condensed together on one side of the nucleus. Moreover, hybridization, using molecular hybridization to fractions from a sucrose gradient, showed that the denser fractions that had a small amount of DNA shown in red hybridized extensively with ribosomal RNA shown in blue. And this was at a level much higher than had been published for somatic nuclei, and therefore suggested that the ribosomal DNA was amplified in the Xenopus oocyte. In fact, the same conclusion had been reached the same year by Don Brown, working together with Igor Dawid at the Carnegie. And here you see the beautiful GV with the amplified nucleoli, which contain rDNA. So using that as a model system, Joe then was able to succeed in working out the initial details of in situ hybridization. He then went off for a sabbatical in the UK, where he continued to work out the details of this. And meanwhile, my classmate, Mary Lou Pardue, was working on her thesis project, which was to ask if the rDNA was excised from the chromosome or was it copied from the chromosome to form the extra chromosomal amplified rDNA in the oocyte. This was technically very challenging. Mary Lou is incredibly clever, and she persisted in trying to solve this problem using lots of elegant approaches, using a lot of ultracentrifugation, but the project was stymieing her. While Joe was away on sabbatical, Mary Lou was doing a centrifuge run where, unfortunately, the SW25 rotor blew up in the ultracentrifuge and destroyed both the rotor and the centrifuge. The reason for that is that there was a tool that none of us knew about that had come with the SW25 rotor, where you were supposed to tighten a screw at the base of the rotor before using it each time. And of course, none of us had done that, and it was just Mary Lou's poor luck to be the one using the rotor when it finally gave out and came off the shaft in the centrifuge. So the question was, should we notify Joe, who was on sabbatical, about this mishap? And we decided we did not want to ruin his sabbatical, so we would not tell him until he returned to Yale. So as soon as he returned to Yale and walked in the door, Mary Lou ran up to him and said, "I have something to tell you that's very important." And then she broke the news about the centrifuge having blown up. Joe invited Mary Lou to switch thesis projects because her project, although an important one, was a very difficult one, and to work instead with him on refining the details for in situ hybridization. And as was published by them in 1969, and you've already seen this picture from Joe, the results worked because Joe was a biologist and had used the best biological system to answer the question. Meanwhile, it was important to also see if this method of in situ hybridization would work on genes in chromosomes. And for that, I collaborated with Mary Lou so that we would look at the gigantic polytene chromosomes of the fly that I was working with, Sierra, which I had brought with me from my undergraduate days where I had done my senior honors thesis with this fly. And Joe had convinced me that rather than work on Lampbrush chromosomes, I should continue my work with Sierra, which I'm still continuing today. And so we used ribosomal RNA as the probe for in situ hybridization to polytene chromosome squashes, and the results were lovely. And here you see the very first in situ hybridization to a chromosome, which was part of my thesis, and also we published in 1970 as a group, and subsequently I published in 1971 as well in a follow-up paper. Now it's important to note that in situ hybridization continues today as a really important method, especially for its use in clinical applications, where it's used for many diagnoses. At the time that Joe developed the method, it was not yet common to patent research findings. And so no patent was filed by Yale for Joe's finding of development of the method of in situ hybridization to identify genes and chromosomes. Of course, today, he would probably be a billionaire if a patent had indeed been filed because of the importance of this method. Meanwhile, at the time that we were working out the polytene chromosomes, Mary Lou was looking at a different probe, namely that from mouse satellite DNA, to see if that also would work on chromosomes, and indeed it did, lighting up the centromeres of all the chromosomes. She also was trying to do the same experiments using satellite DNA from guinea pig, and a guinea pig was brought to the lab to be used for these experiments. However, this became a lab pet, and this guinea pig, whose cage was near our lunchroom, knew everybody's footsteps and knew who was going to come and feed it by simply who was walking down the hall, and it would make little squeaking sounds. It was especially fond of Ed Cohen, who fed it large amounts of apples. So we were all worried that this guinea pig was going to become satellite DNA from Mary Lou's experiment, but then at the time, Ken Jones was also getting ready to publish similar work about mouse satellite DNA being used for in situ hybridization. And so Mary Lou figured that, and Joe figured that they better not dawdle along in looking at guinea pig, and so they simply published the paper just with mouse satellite DNA, and the guinea pig went to the home of some family that was happy to have it as a permanent pet. Well, as you've already heard, Joe is a biologist extraordinaire. He's worked on many diverse organisms, some of which are shown here. And here you see a picture of Joe collecting some organisms in the wild. And let me conclude with an anecdote about a search for the red eft, which is the juvenile stage of the red-spotted newt. I was at a Gordon conference as a grad student, and Joe Gall and Oscar Miller were going to take a hike looking for the red eft and asked if I wanted to join them. So I was happy to do so, and we went traipsing off through the woods, and they were meandering along, looking under every branch and tree stump to see if they could find stone, to see if they could find a red eft. Meanwhile, I was getting eaten alive by mosquitoes, and those of you who've gone to Gordon conferences know that there are a lot of mosquitoes in New Hampshire in the summer. And so I started running ahead on the trail because I didn't want to get more mosquito bites. And in running ahead to try and get out into the meadow where it was sunny and there were fewer mosquitoes, I stumbled over a log. And so Joe and Oscar came running to see if I was okay. And instead of paying attention to me, having stumbled, they looked with delight because I had overturned the log where there was the red eft, which you see here. So much for adventures with Joe. It's been a wonderful treat to have had him as my Dr. Vater, which is German for Dr. Father. And we thank him for his spectacular career and congratulate him for a spectacular career full of scientific discoveries. and I send him and Diane my very best wishes for a very happy retirement, including continued microscopy to view his favorite slides. Thank you very much.
Thank you, Susan, for that wonderful talk. Our next speaker is Sir Adrian Bird, who's going to be talking about ribosomal DNA, or rDNA, structure and amplification. Adrian did his PhD with Max Bernstein at the University of Edinburgh on amplification of amphibian ribosomal DNA, which you now know was a subject very dear to Joe's heart. He came to Yale as a postdoc in Joe's lab in 1971. Here he continued to work on rDNA in a wonderful collaboration with two fellow postdocs, Jean-David Rocher and Amy Bakken. He used EM autoradiography to demonstrate that amphibian rDNA was amplified by a rolling circle mechanism. Adrian has spent most of his career at the University of Edinburgh, where he helped set up and served as director of the Wellcome Trust Center for Cell Biology, and where he is now the director of the Simons Initiative for the Developing Brain. Adrian's research focuses on DNA methylation. He identified CPG islands as gene markers in the vertebrate genome and pioneered the discovery of proteins that read that DNA methylation signal, such as MECP2, whose mutation causes the severe autism syndrome, Rett syndrome. Like many of Joe's progeny, Adrian has a long list of honors, such as membership in the Royal Society and the US National Academy of Sciences. But I believe he is the only former Gall lab member to be knighted by his queen for his services to science. Adrian.
Hello, am I audible?
Yeah, we can hear you. There you go.
Okay. Thank you very much. It's a great privilege to be asked to contribute to this important meeting where Joe is semi-retiring. I would like to try and transport you back to chromosome biology in 1971, which I believe is the year in which I joined Joe's lab. It might have been 1972, I'm not totally sure. Some questions that I encountered when I got there, which I had not really thought about before, because they were discussed very often at lab meetings, were how many genes were there? Do Drosophila polytene chromosome bands signify genes? How many copies of each gene? There was the master-slave hypothesis, which is probably not a title one would give to a hypothesis these days, but this was obviously an attempt to try to understand how many genes there were and how they might correct against each other. And Lampbrush chromosomes were invoked there by Mick Callan, whose hypothesis this was. Do all cells in the body have the same genome? This wasn't totally clear, and that's why gene amplification threatened to be a paradigm for development in some respects. Chromosome structure we've already heard about. There was unanimity was established both from genetics and also from Herbert Taylor's experiments that Joe has championed over the years. And Joe's DNA is one experiment which we've heard about. There was also Duport's folded fiber model, which I remember people talked about a lot. I found it difficult to get excited by it, but and I think it has receded into history now. Particularly now we know more about the structure of chromosomes. There are still questions there. We didn't know what a centromere was. I mean, and a lot of people still work on that. We didn't know what telomeres were. In a way, they're the organs of chromosomes. How do they work, and origins of replication as well. So the knowledge was quite primitive, but some of these questions you will recognize still persist today, including this one. What are Lampbrush chromosomes really doing? Although it's become pretty clear that they're and what they're doing. So my brief is supposed to be gene amplification. We've already heard quite a lot about that. I'm going to rehash some of it again, but I will stray outside Lampbrush chromosomes a little bit, perhaps, and I hope you'll forgive me for that. These are Oscar Miller's Christmas trees. Oscar Miller, Joe's first postdoc, although he was older than Joe. And these beautiful pictures never cease. I know we've seen them several times, never cease to engage the attention. For me, what was amazing was that all the structure of ribosomal RNA genes that have been deduced by sonication and physical methods and hybridization were spectacularly vindicated by this one picture, even down to the spaces between the genes. This was already inferred by Don Brown's lab and by Max Bernstein's lab from their indirect approaches, no restriction enzymes in those days. So if you had to draw a cartoon of what ideally transcription of ribosomal RNA genes would look like, this would be it. Then I just couldn't help adding that because of Joe's long-standing interest in Lampbrush chromosomes, I prefer the German Lampbrush and Chromosomen. It's got more gravitas, but these also have pictures. This in the center is a picture of a loop from I believe it's nodophthalmos, showing the polarization of lengths that is explained by the Miller spreads. Ulrich, this is an example. I'm not giving this in the chronological order in which things were discovered, but it shows this amazing transcriptional elongation within these loops that explains the polarity. And I think Joe's sort of synthesis these days of what's going on is that the oocyte is such a large cell that the need to fill that cell developmentally to achieve what eggs achieve, and also the turnover of proteins that is constantly going on, require the oocyte to transcriptionally run, if you like, in order to stand still. And this transcription is a sort of almost clumsy attempt to desperately keep up the levels of proteins in those cells. So that's nothing to do with amplification. So I now need to come back to amplification. Here we have the paper that's already been referred to by Joe. It was published in 1968 alongside the paper, or at least at the same time as the paper from Don Brown and Igor David. And I just noticed that it's got these two dots over the second O. And maybe I'm wrong, but it seems to me people, in the absence of this diaresis, do tend to say oogenesis more and oocyte more than they used to. So I think we should campaign to have the diaresis restored. Anyway, Xenopus sometimes has a reputation for being ugly, but as you can see, this is completely wrong. It's a beautiful looking animal. And of course, in those days, as Don Brown has cogently described in some articles over the past few years, only one or two types of genes could be studied, and they were multi-copy genes that could be isolated by physical methods. And ribosomal RNA genes are amplified thousands of times, as we've heard, in amphibian and fish eggs. And it happens at this stage when this beautiful tadpole the ovary is entering Pachytene stage of, and this is a rather poor reproduction of the figure that Susan showed a very nice example of, showing that this peak on the left, the peak on the left, which would represent normally 0.2%-ish of the total genome, is spectacularly labeled by tritiated thymidine, meaning it's replicating independently of the rest of the chromosomes. So this was amplification. You've seen this picture before. This is Feulgen stain, showing that the molecular biology in the form of the cesium chloride gradients is matched by the cytology, which is a classic Joe Gall approach. And here's the in situ hybridization validating that this is a vast accumulation of ribosomal RNA genes. And Ginger referred to these experiments. And unfortunately, I was unable to capture a photograph of any kind of Amy Bakken, but she was involved in this, this collaboration as well. Jean David knew how to do electron microscopy of DNA, and so we did autoradiography. So and this shows an example of a DNA molecule snaking around here with a circle at the end, and basically this is a replication fork traveling round and round this circle, and we showed that really by doing a pulse chase experiment where the pulse chased the labeling in the tail further and further away from the circle in a way you would expect if it's a rolling circle. So the salutary lesson was that I had been working on this in Joe's lab and had formed the conclusion that replication of these genes was actually by bidirectional replication in the way that Huberman and Riggs had shown in their experiments using fiber autoradiography. And I'd written a manuscript and I was about to submit it. And then Joe, I think it was Joe, went to Harvard and met David Dressler, and his lab had found lariats, namely these things, in amplifying ribosomal RNA genes. And I knew immediately, I was incredibly depressed for at least a day. I knew that they were right and that I was wrong. And I've always kept this manuscript because it showed me how easy it was by selecting your images or your data to delude yourself, because actually the rolling circle was correct. David Dressler's lab had got the right answer. And the Route 66 business is because I remember saying to Joe, Joe, I'm going to, with enormous entitlement that was unjustified, I'm going to go buy a car and drive to Los Angeles and then drive to Vancouver and then drive back along the Trans-Canada Highway. And Joe said, how long is that going to take? And I said, about 6 weeks. And Joe said immediately, oh, really? Whose lab are you coming back to? So I realized at that point that it wasn't something I could necessarily assume. Anyway, I went, and Joe, because he allows an enormous freedom to people who work in his lab, indulged me. And when I came back, we were just developing these autoradiographs here, which vindicated the approach to proving that the lariats were the intermediates in replication. So amplification, frogs do it, fish do it. This is a picture taken from Joe's book of visualizing cells, showing the nucleoli. And actually, Theophilus Painter speculated, and this I got from Joe's book, that these were independently replicating nucleoli that had come out of the chromosomes, and he was completely right. And then water beetles do it. This is Jean David and Joe's amplification paper showing a spectacular accumulation of ribosomal RNA genes. And you remember, obviously frogs do it, fish do it. This is a reference to Cole Porter, only in his case it was birds do it, bees do it, but actually birds and bees almost certainly don't do it because they rely on nurse cells or other cells to fill up their oocytes with goodies. But the third line in Cole Porter's song is educated fleas do it. And this is a picture again from Joe's cell biology book, a picture of a flea done by Robert Hooke. I don't know if fleas do it, but Joe almost certainly does know if fleas do it. I'm not sure what sort of oogenesis they have. And then we've heard about tetrahymena. It was coming into the lab just about the time when I was there in 1971, 73, or 72, 74. And it has this amazing micronucleus, macronucleus structure. And Kathy Carr was the one who showed, actually, while Joe was on sabbatical in the UK, that it had this palindromic structure, and she wrote to Joe a palindromic message, which he was initially confused by, but then managed to understand. And then he replied also in a palindromic message. You're probably going to ask me what were those palindromic messages, but I don't actually know what they were. It's quite possible that when the anecdotes are written out, then those anecdotes will accompany them. Anyway, on the right here is a picture of some of the amplified ribosomal DNA from the macronucleus, self-associated, it's melted, and the one on the right is self-annealed. So in other words, it's a hairpin that has snapped back as proof that this is a palindromic structure. And you can see that the one on the left is the molecules are thinner and more kinky in appearance. So in those days, gene amplification was not only fashionable, it was bordering on kinky. Finally, something that is a bit of a cheat to include, but it's a sort of bijou study that I think is a beautiful little study, rounded, rigorous, and it was going on about the time I was in Joe's lab, and that is the alpha heterochromatin, the unamplification of alpha heterochromatin, because the rest of the polytene chromosomes are replicated about 2,000 times and stacked up to form these beautiful structures in the salivary glands. But the chromocenter, and this is true even in Drosophila virilis, where half of the DNA is satellite DNA, is rather shriveled and small. And Joe characterized along, I should mention Emil Heitz here, who has the dubious honor of having coined the word heterochromatin. But the important thing is that he recognized what heterochromatin was and euchromatin was, and he gave them these rather cumbersome Greekish names. But the alpha heterochromatin, Joe speculated wasn't being amplified, and this is a melanogaster on the left and virilis on the right, and you can see here again the combination of cytology and genetics. Cytology and molecular biology, sorry. This is the satellite in melanogaster. These are the satellites which make up a huge fraction of the genome in virilis. And then the salivary glands, they're not there. And that's because this region of the chromosome is not amplified when these polytene chromosomes are formed. So I'll finish up just by mentioning, and I'm rehashing some things here, Joe's a unique approach. How on earth does he do it? Keep on making these contributions. He uses his knowledge of biology to find informative outliers. So he knows enough about biology to know where the sort of sensitive points are, where he might be able to learn new things. In a sense, Lampbrush chromosomes are that, tetrahymena is that, polytene chromosomes are as well. He combines microscopy with molecular biology. In fact, I now notice from that cutting that Liz Rogers showed that was the reason why they wanted him in Yale, because in those days, a microscopist did microscopy and molecular biologists did their thing. And Joe realized that by putting these together, he could do what he actually wanted to do, which was answer questions. Experimental rigor, and this, my experience in his lab where I deluded myself, was sharpened by the fact that I was surrounded by a intellectual climate that regarded truth as a sacrosanct. And obviously, in these days, that is ever more true. It's a lab of equals. It was a lab of equals, and that's Joe's style. He is not a co-author on any of the papers that I was involved in from his lab, and we've heard of other examples of that. From Jones, for example. And that's symbolic of the way in which he gives freedom to the people he works with, and is not feel that their performance is part of the way in which he will be judged. Clear thinking. and presentation. And the word I came up with is, that comes to mind anyway, I didn't come up with it, is limpid. It's, if you read the way in which he writes, it is simple, it is clear, and I think it's clear presentation and clear thinking go together. Infectious enthusiasm. He has trained so many people because he himself is desperate for this knowledge, not desperate, but wildly enthusiastic about getting this knowledge, and that is communicated to anybody who's exposed to him for any length of time. And finally, on my list, and you can never reduce anybody to a list of bullet points, obviously, he's aware of the history. He knows that he is part of a cultural plant with its roots in the Renaissance, really, where people have been accumulating knowledge about the world, our place in it, and those who cohabit it with us. And this is the book that I've referred to a couple of times, which has lovely stories, informative information. So Joe, thank you very much for, I thank my lucky stars that I had the chance to work in your lab. It is something, and I think I speak for many people, that, and obviously it's been said before, something that I have carried with me. Those values I have attempted to emulate, and I wish you luck in the next phase of your semi-retired career. And that's the end of my presentation.
Thank you, Adrian. Okay, our next speaker is Liz Blackburn, who's going to be speaking about heterochromatin and repeated DNA. Liz was raised in Tasmania, Australia. She did her PhD with Fred Sanger at the MRC Laboratory of Molecular Biology in Cambridge, where she was part of the pioneering work that led to the sequencing of the DNA genome of Phi-X174 bacteriophage. When she came to Joe's lab in 1975, she was one of only a few people in the world who knew how to sequence DNA. She told Joe she wanted to sequence something important. This was before cloning, so Joe suggested that she sequence the ends of tetrahymena ribosomal DNA, which he had just been able to recently isolated by use of cesium chloride centrifugation. The telomere sequencing work was described in a 1978 paper by Blackburn and Gall that began the molecular era of telomere biology. In her own lab, Liz continued to work on telomeres, first at Berkeley, where she and her graduate student, Carol Greider, isolated and discovered telomerase, the activity that lengthens the ends of chromosomes in almost all organisms, and then at UCSF. Her pioneering work on telomeres has been recognized in innumerable ways, including with the 2009 Nobel Prize in Medicine, which she shared with Carol Greider and Jack Szostak. Joe was her guest at the Swedish award ceremonies. Liz.
Thank you, Ginger. So first, my warmest congratulations and best wishes to you, Joe, on your retirement. And we look forward to the science that we know is still to come from you despite what is called retirement. And this is just a wonderful opportunity today to celebrate all your great science that has happened. So my assignment for this talk, as Ginger mentioned, is to briefly highlight research into heterochromatin and particularly simple repeated DNA sequences in Joe's lab. And although I can only include just a couple of the wonderful reminiscences that several of you involved in these studies sent me, don't worry, because Ginger Zheng is compiling the full set of great stories and reminiscences that various of us have been collecting. And so Ginger will be sending the whole thing to you. So I'm going to start by reminding you, though, of what Adrian Bird alluded to. And that was that Joe is a superb scholarly historian of science and witness this marvelous book that Adrian alluded to. And in here, you can see just one of the collection of plates illustrating this that consists of original photomicrographs that are taken by Joe himself. And so I've put it into my background. Can you see here? This is the plate where you can see on the top. The important point here is that it relates to Joe's area of long fascination, which is highly repetitive DNAs, and many of which you've heard about, and ribosomal RNA genes, for example, but many of which are satellite DNA sequences, as we've been reminded that you can get by cesium chloride or cesium sulfate density gradient centrifugation. And on top, you see the darkly staining regions of chromosomes in Drosophila melanogaster and D. virilis, and those are the heterochromatic regions. And as Joe had shown very clearly with Mary Lou Pardue in mouse initially, clearly the satellite sequences were located in the heterochromatin. And below, just to remind you again of the point that Adrian made, was that satellite DNA was underreplicated in the Drosophila polytene chromosomal heterochromatin. So it was through the early 1970s, Joe went on multiple really fascinating explorations of simple sequence satellite DNAs, as well as the other repeated DNA sequences mentioned, and especially with lab members I'll mention: Ed Cohen, Diane Atherton, Mary Lake Poland, Ronald Eckhart, Sharon Endow, Ginger Zakian. They addressed fascinating and important biological questions about satellite DNAs, their biophysical and DNA sequence characteristics, the latter of which I'll refer to a little bit more, their replication patterns, their amplification, their chromosomal locations, sometimes their extrachromosomal nature, as particularly for the ribosomal RNA genes. By the way, Joe in recent years hasn't forgotten simple sequence satellite DNAs, as witness his 2016 paper on genome evolution of multiple Hawaiian Drosophila species, particularly their various expansions of satellite DNAs. So even as recently as 2016. Now, how I came into the world of these simple repetitive DNA sequences was because they turned out to converge with another fascination of Joe's then, which was ribosomal RNA gene amplification. And so back in 1974, as I was completing my PhD in Fred Sanger's lab in Cambridge in the UK, and their new methods for RNA and then DNA sequencing were being developed, they were using prokaryotic systems. And I was greatly impressed by Joe's ready development and adoption of always the latest and greatest techniques to address interesting and biological questions. Particularly, I was struck by nucleic acid sequencing, and he was doing it in eukaryotes to boot. And here's another thing that absolutely riveted me. It's right behind me, and now I'll move over so you can see. It is the image, another image from Joe's 1974 PNAS paper, showing the amplified tetrahymena rDNA and showing it was in the form of many copies of extrachromosomal short, linear, apparently identical 20 kilobase pair rDNA molecules, and some fraction of which, you can see one here, could be found as circles under the electron microscope. Now, why did this rivet me? Well, it was because this DNA form was superficially like that of the linear bacteriophage lambda DNA, which could form circles via its cohesive complementary DNA sequence, single-stranded DNA ends. And the very end sequences of those linear 50 kb lambda bacteriophage DNAs had originally been laboriously, I might say, sequenced by Ray Wu and Dale Kaiser in 1968 using repair reactions by DNA polymerase, and by Ken Murray extending with more methodologies in 1972. And this really excited me because back then there weren't many ways to get directly at DNA sequences, apart from copying them into RNA and piecing together the RNA sequences, which Joe and his lab had actually done in their pioneering satellite DNA repeat sequence characterizations, and I'll allude to that a bit later. So I thought, okay, well, Okay, it had been possible to get an end sequence for 50 kb, lambda-phage DNA, and here Joan succeeded in preparing homogeneous populations of an even shorter, a 20 kb DNA, the tetrahymena rDNA. And it was critical and characteristic of Joe's acumen about the right biological system to use, that Joe strongly urged me to use these tetrahymena rDNA linear molecules for my DNA terminal sequencing effort, rather than the even shorter oxytricha macronuclear linear DNAs that I had briefly contemplated for this purpose. So, as so often, Joe made the absolutely right call. And the icing on the cake when I joined Joe's lab was that Kathy Kara had just demonstrated that the rDNA molecule was palindromic. So the two ends might even be identical, which would make it even more technically attractive as a DNA whose ends could potentially be sequenced. So extending Wu's and Murray's leads with lambda DNA, I could then stitch together a terminal sequence, and I added in some additional techniques for DNA sequencing determination that had been developed in Fred Sanger's lab group. So it was in these tetrahymena rDNA molecules that Joe's interest in rDNA amplification and repetitive DNAs converged, in this case in the repeat sequences that turned out to be the simple sequence telomeric C4A2 repeats that I was able to sequence first at the ends of this rDNA, and then still in Joe's lab, Meng Chao Yao and I were able to show that C4A2 repeats were also at the ends of the processed non-rDNA macronuclear genome linear DNA molecules in tetrahymena. So I just want to reflect briefly on the methods that Joe and his lab members brought to bear on repetitive eukaryotic DNAs. It was just completely characteristic of Joe, as we've heard, that he was a very inventive developer and earlier adopter, in this case of the newest of molecular and biophysical techniques for nucleic acid sequence analyses. But we've also seen another part of Joe, and I'll just allude to what he says in his autobiographical memoir, Chromosome Odds and Ends, which is published in Annual Reviews of Cell and Developmental Biology. And Joe, in describing his childhood, says, "In retrospect, I realized that most of my knowledge of the outside world was coming through my eyes, and that has been the story of my life." End quote. And we've all seen the beautiful demonstrations that Joe's scientific evidence is often highly visual. Just one great example is the in situ hybridization of satellite DNA to heterochromatin regions of chromosome spreads. It struck me also how visual, in a different way, was the first data from his lab that some repeated sequences were actually very simple sequences. Here's Sharon Endow's anecdote, and she talks about how an audience reacted in the very early days of DNA sequencing. Sharon said, "My first conference talk was at the Gordon Research Conference on nucleic acids in 1974. When I showed my sequence data, there was a low murmur across the audience, as there were only 3 or 4 spots, since my data were for simple sequence satellite DNAs, by contrast to the promoter and other sequences that the other conference attendees were reporting." And so Sharon's spots were of a 2D fractionation of fragments of P32 labeled RNA copies of DNA. And in fact, it turned out that they're able to show that the D virilis sequences that were the satellite sequences being analyzed consisted of 5 and 7 and 10 or 12 base pair, various kinds of base pair repeat units. And tetrahymena telomeric sequences also revealed their simple sequence and highly repeated nature to me as a major single spot this time on a 2D fractionation of P32 labeled products. In this case, it was from cleavage of depurinated DNA, which led to a prominent C4 spot on an autoradiogram, showing that they contained a purine CCCC purine sequence that was present in many copies. And Ed Stevenson, who was there at the time, wrote me recently, I quote, "I have a vivid memory of Liz wandering the 4th floor Klein hallway with an X-ray film, still wet from the wash tank, and explaining to anybody who would listen the experiment that showed that tetrahymena telomeres are composed of the sequence C4A2. So I just want to finish by pointing out that, you know, in Joe's lab, we all cared about the success of all of Joe's projects. Let me tell you a little incident about newts. So once I volunteered to go newt hunting in the woods near New Haven to provide animals for Joe's Lampbrush chromosome studies. And funnily enough, like Sue Gerbi's My Animal Hunting Story is also a newt story. So I was proudly armed with a styrofoam collecting box. It was a hot, humid day. And I must say, you know, one newt looks pretty much like another to me. Anyway, I sweated mightily as I captured one newt after the other. And as I raised the lid to put the next one in, the one I just caught would jump out. So it was a long, sweaty hunt. You know, I was pretty incompetent. But I ended up with, you know, a grand total of just a few. So I proudly brought them into Joe's lab. And he opened the box and he said, "Oh yes, that's the fast species." But I was closed with two important things that have stayed with me from being in Joe's lab, because I've heard them from so many other past members of Joe's lab too. We've heard them today, and people writing to me recently told me so many times, over and over. First, Joe's supportiveness and kindness to everyone in his lab, and the spirit of good interactions among the lab members. And second, when encountering some lab or research-related situations, I heard this over and over. We always think, what would Joe do? So I want to join every one of us in saying, I'm so grateful to you, Joe, for so much. Thank you.
Thank you, Liz. Our next speaker is Giuseppina Barsacchi, who will talk about Lampbrush chromosomes. Giuseppina was educated at the University of Pisa, where she became a faculty member in 1969. In the first part of her career, she studied amphibian Lampbrush chromosomes, and because of this shared interest with Joe, she came as a visiting scientist to his lab in 1969. Giuseppina and Joe must have had a really great time on that first visit, because Giuseppina came back 4 times to work in Joe's lab. In 1990, Giuseppina set up the developmental biology lab at the University of Pisa and switched her research to the development of the vertebrate eye. She's held multiple important positions in her university and in European science. Among her many honors, in 2005, she was elected to the Italian equivalent of the US National Academy of Sciences. Although this organization was founded in 1603, Giuseppina was the first female member of the Academy Council. Giuseppina.
Hello to everyone and good morning, Joe. And I wish first of all to thank the organizers who thought about this beautiful meeting and also worked to make it a success, as we heard already beautiful things from Joe first and from all the speakers afterwards. So thank you to all the organizers. I wish to, I thank the organizers for having asked me to talk about Lampbrush chromosomes. But I thank Joe, Liz Rogers, Susan Gerbi, Adrian Bird for having laid down the hard science about Lampbrush chromosomes. And I will mostly confine my talk on how the Lampbrush chromosomes mediated my meeting with Joe Gall. And it happened that my supervisor, here looking for newts, was interested in amphibian karyology. So he went to spend some time with Mick Callan in St. Andrews. And he learned there how to work, how to make Lampbrush chromosome preparations, so that when he came back in Pisa, we began studying Lampbrush chromosomes of many newt species, and we also drew them ourselves, as you can see here. And I began reading the pertinent literature. And here is where I met for the first time Joe Gall in this review paper that has already been quoted, this 1954 paper. I truly admired Joe's work and the way he wrote about his work. And I remember I thought, well, if this man resembles his writings, then I'd like sometimes to work with him, maybe. But then when I won a scholarship to visit the States for 1 year, with my supervisor, we thought that maybe it was too much of a jump for me to go to Yale immediately. And I started by working with a very nice scientist and gentleman, Alan Humphreys, on oocyte maturation. And Lampbrush chromosomes were still, of course, involved. In fact, I had the chance to visit Oscar Miller in Oak Ridge. I was close to Oak Ridge at that time, and he taught me how to make his famous Miller spread. But I continued to read and study the Joe's papers. So when I felt more confident about myself in the States, I wrote him a letter saying, well, I'm interested in Lampbrush chromosomes. I am here, I'm doing this and this. I would like to visit your lab. Can I come? And I thought maybe he will not answer to me. I thought it was probable that I was not going to receive any answer. And instead, Joe called me back. So you can imagine, I was so surprised and excited because after such a long time reading his paper and his work, I was eventually talking with a real Joe Gall. So during that conversation, we arranged for a visit, for my visit to Yale. And I went there and I spent 3 months in the famous 4th floor of the Klein Tower. And I met many interesting and good people. And it was a very exciting time there, as other speakers mentioned. It was a time of ribosomal DNA amplification, of in situ hybridization. And I was also introduced to the famous Model ultracentrifuge. I fell in love with it. Okay, well, it was a mixture of love and hate because when I was running it overnight, I had nightmares of the centrifuge exploding. But in any case, we were able to isolate a satellite DNA component from the newt, which was located at the centromeres and sphere loci by in situ hybridization. You can appreciate here, however, that the morphology of these Lampbrush chromosomes isn't great. Whereas when I went again during one of my successive visits to Yale in 1979, this was the morphology of the chromosomes that we obtained in the lab after in situ hybridization with different probes. Just beautiful, just beautiful. And the miracle was obtained by centrifuging the Lampbrush chromosome slides on swinging platforms in swinging buckets rotor. And the platforms were held by rods that in the first version of these devices were made of aluminum. But not everything went smoothly. In fact, at the very first test, we all were around this Sorval centrifuge, and Joe started the centrifuge that reached 1K, 2, 3 K RPM. Everything was smooth up to that point. Visitors came to meet Joe, so he left the lab and went to meet the visitors in his office. And the centrifuge continued running up to 5K. At that point, we heard a terrible noise, a huge, horrendous noise, and the centrifuge began to move around a little and jump. And we, of course, stopped everything, and we saw the Joe's face at the window between his office and the lab, and he sort of grimaced a little, and then he went back to restitute attention to his guest, displaying an admirable self-control. So this centrifuging was done on a project of Joe, an idea and the project of Joe, and the devices were made again, but with rods made of steel this time, and centrifuging went very well in different experiments, but still the Lampbrush chromosomes were not satisfactory. They were somehow distorted, and Joe wanted to understand what was the reason, as you can see in this cartoon made by Manuel Diaz, and realized that the devices never went completely horizontal, but they remained with an angle, and so the slides were never perfectly vertical, and therefore he sought to make, as he writes in this autographed letter, near perfect Lampbrush chromosome preparations. He thought of introducing a supporting wedge under the slide. This is his own drawing. So to have chromosomes beautifully displayed, and that's what he did. And from that on, we had wonderful Lampbrush preparations thanks to the centrifuging technique. I think this shows the extreme care Joe has for all details, technical details, cultivating the big issues of science. And centrifuging Lampbrush chromosomes, of course, reduces their complex three-dimensional structure to 2 dimensions, so that Joe and his colleagues were able to analyze the transcribing loops by super resolution imaging, and to propose models of transcription units that have a bear on the way we think about transcription in general. Before concluding, I would like to remember 2 close friends of Joe, and also of a number of us, of many of us, Herbert McGregor and Mick Allen. I would say 2 Lampbrush friends of Joe, and I was lucky to overlap in Baltimore with Mick and Amarilde Scallan in some of my visits, and we had a very good time together. And as you know, Joe and Mick were extremely good colleagues and friends, even though they had different personalities. And in fact, it was some of a problem that Mick used to smoke in the lab. And I remember this authoritative or imposing person, such as Mick was, hiding his cigarette behind his back while meeting Joe in the hall, like a child caught with his fingers in the jam. So in the end, I wish to show the acronym I dedicated Joe for his 90th birthday, which says just, only, in quotes, science, ethics, purpose, heart, generously, give soul, live life. So thanks, Joe, for everything, and best wishes to you, your family, and everyone. Thank you to everyone.
Thank you, Giuseppina. Our next speaker is Zhilong Liu, who's going to be talking about Cajal and other nuclear bodies. Zhilong is also a co-organizer of this meeting. He received his PhD from the Chinese Academy of Sciences Institute of Zoology in 2000. He was a postdoc in Joe's lab from 2003 to 2007. In Joe's lab, he identified the long sought after Cajal body in Drosophila. He must be a discoverer at heart because while he was doing his postdoc, he identified two additional subcellular structures: the chambers or nuclear histone locus body and the cytoplasmic U body. Zhilong set up his first lab at the MRC Functional Genomics Unit at the University of Oxford. Here he discovered yet another new structure, the filamentous cytoophodium, and showed that it contained CTP synthase. In 2016, he moved to Shanghai Tech University, where in addition to being a professor, he is also a vice dean. His current research focuses on how the assembly of metabolic enzymes into specific structures affect metabolic regulation. He still collaborates with Joe. Zhilong.
Am I on?
Yep, we can see you and hear you.
Good afternoon, Joe. Hello, everyone. This is Zhilong. It's midnight in Shanghai. It's my great honor to speak here to celebrate Joe's remarkable career with astonishing discoveries. In the next 10 minutes, I'm going to introduce 7 theorems of Joe. Amphibian oocytes are big. The GV, the nucleus, are huge. So you can dissect them by hand, just as Joe did. In 2003, Corey and Chris published a paper in JCB showing the Cajal body components can come from nucleoplasm into the Cajal body dynamically. That year, Sveta, a girl from Russia, joined Joe's lab. Her question was to see if Cajal body components can come out from the body. She used photoactivable GFP to do the experiment.
You can't see your PowerPoint if you're supposed to be showing anything right now.
Oh, no. Oh, you cannot see. Oh, okay. Let me see.
Go to share screen at the bottom.
Oh, sorry. Oh, okay.
It's okay.
Let me do it again.
Yeah, start over.
Start over. I will be very. I thought you already see my PowerPoint.
And then make sure that's full screen. There you go. Yep.
Is this so you can see it, right?
Yeah.
So yeah, this poster, this supposes a mod for Joe's 85th birthday. I just changed the title. And the next one, that's the title of my talk today. So I want to introduce 7 theorems of Joe. I talk about the GV, that's huge GV, because they're so big, so you can dissect them by hand. That's Joe always did. So in 2003, Corey and Chris published a paper in JCB showing Cajal body components can come in from nucleoplasm to the Cajal body dynamically. So that year, Sveta, that's a Russian, joined Joe's lab as a postdoc. Her question was to see if Cajal body components can come out from the Cajal body. She used photoactivable GFP to do the experiment, but nothing happened. Then she decided to repeat Corey's experiment. She couldn't. She couldn't repeat Corey's experiment. She was so frustrated. She asked Joe. Immediately, Joe realized mineral oil was kept in a plastic container, which makes mineral oil toxic. So a new batch of mineral oil in glass bottle solved the problem. Next year, Sveta and Joe published a paper in PNAS showing dynamics of cooling in Cajal bodies. So, theorem number 1, if you have trouble, ask Joe. On the first work day of 2003, I joined Joe's lab. One question in the lab was, does Drosophila have Cajal bodies? It was difficult because the signature protein coilin has not yet identified in Drosophila. People started to believe there's no coilin in Drosophila at all. But luckily, U7 and U85, this RNA is supposed to be a marker for Cajal bodies has been identified in Drosophila. And the strategy was to use RNA instead of a protein to identify the Cajal body in Drosophila. So I started to learn in situ hybridization, which was invented by Joe many years ago. To make a long story short, eventually I got it done. I got U7 showing 1 dot in the nucleus. I got U85 showing 1 dot in the nucleus. Joe said, perhaps you can see if these 2 dots co-localize, then we'll be convinced. I did. But you see, 2-color fish of U7 and U85 shows 2 dots in 1 nucleus. Sometimes they're close together, but many times they're far away. I remember that's Friday afternoon. I talked to Joe. I said, Joe, I don't know why. There are 2 dots, not 1. I was so disappointed. But Joe was very excited. He said, this is unexpected. This is something interesting. So he encouraged me to do the following experiment using different markers. Eventually, we realized that there are 2 separate bodies in the nucleus. One we decided to keep the name Cajal body, and the other we named as a histone locus body because of its association with histone gene clusters. In addition, we found the 3rd one in the cytoplasm. We call it U body. The next year or 2, Joe and I, and also together with colleagues, published 2 papers in JCB and PNAS describing these findings. So, theorem number 2. If you are disappointed, talk to Joe. We'll also look into nuclear bodies in Drosophila GV. Alison Singer just got her bachelor degree from Brown University, recommended by Susan Gerbi, joined Joe's lab. Encouraged by Joe, Alison looked into the GV and found inducible nuclear bodies there. You can see even needles there. They published a research paper together. The next question was, does Drosophila have choline? I remember the last 2 days of 2005, using a new strategy, I found a putative hit, only one out of several hundreds. On the first work day of 2006, I talked to Joe. I said, I found one putative hit of Drosophila choline, but I was skeptical. Joe said, this is something suspicious. We should go for it. Encouraged by Joe, we did transgenic fly, which fused this putative choline to GFP. When the fly was available, Joe and I looked at them and found the bright dots in the nucleus. Immediately, we realized that we have identified choline in Drosophila. In the following years, collaborated with Dana Carroll, we used zinc finger nuclease to generate mutants for choline. This was one of the first genes in Drosophila being targeted by zinc finger nuclease, leading to 2 publications in PNAS and molecular biology of the cell. So, theorem number 3. If you have doubts, Joe pushes you. Then we have an issue of 2 bodies, the Cajal body and histone locus body. Now Zera and Sweater decided to look back into amphibian oocytes. The conclusion was the choline positive bodies in late stage xenopus oocytes should be considered HLB, that's histone locus bodies, not CB, not Cajal bodies. And then Zera looked early stage oocytes of xenopus and found that a third type of structure that has choline. Zera and Joe call them pearls because choline stays in the periphery. This is not unexpected. In 2000 and 2003, Joe wrote 2 review articles talking about Cajal bodies have studied for 100 years. But theorem number 4, 100 years isn't enough for CB study, says Joe. Last year, Joe and I were invited by the editor-in-chief of Experimental Cell Research, Andreas Simon, to organize a special issue on amphibian model systems. I dug out my notebook of 2003 and 2004, the first 2 years when I was in Joe's lab as a postdoc. 5,000 plus page notebook. But I dug out the data, analyzed them, prepared a manuscript, and sent it to Joe. 2 weeks ago, Joe sent me back his last edit, version 21. So we found a new structure. I call it C-ball, but Joe renamed it as a C-body. We aim to submit this manuscript later this month. Theorem number 5. In Joe's lab, due date is 15 plus years later. I'm lucky to have been working with Joe in the past 17 years. We have science and we have fun. We celebrate Chinese New Year together. We played with dogs. And sometimes Joe demonstrated how to do pull-ups. In 2007, I just set up a lab in Oxford. Joe and I were sitting in the kitchen of my rented house, testing wine. You see, they have 3 glasses there because the photograph, Dan took the photo. You can see me, I was young. In the next 13 years, Joe and I did another set of experiments together. 2007, Oxford, in my office. 2018, in Joe's backyard, celebrating Joe's 19th birthday. Last year, I visited Joe's lab in Carnegie. You see how I was getting old and older. At the negative control, Joe did not change at all. Early this year, Joe was holding a book, Fruit Fly for 100 Years, a Chinese pop science book edited by Wu Mingdeng and myself. Theorem number 6, Joe does not age. And the last theorem, Joe likes glass. I'll show you the slide. Happy retirement, Joe. Lucky to be your trainee. Thank you all.
Thank you, Sheila, for a talk with a lot of good jokes in it. Okay, our next speaker is going to be Gaëlle Talross, who's going to be talking about RNomics. Gaëlle is from France originally, and she was Joe's last PhD student. She worked with him from 2012 to 2017. One of the things she discovered during her thesis work was that some cis-RNAs, which are the spliced out intronic RNAs, are actually localized in the cytoplasm, and that they get there via the mRNA export pathway. Gaëlle is currently a postdoc in John Carlson's lab at Yale, where she's using insect models to test if non-coding RNAs have a role in the diversification of the olfactory system. Gaëlle.
Thank you very much, Ginger. I hope you can all hear me. Let me share the screen. I'm very honored to, I'm very honored to present a little bit of a snapshot of the last 10 years in Joe's lab and represent the RNomic world. So Joe entered this world in about 2010, which was relatively soon after RNA-seq was made available to most labs. And so this was this was entering into this new era was aided by Eugene Gardner, who was a technician in the lab and brute force all the bioinformatics to the lab. So the team has changed quite a bit throughout this last decade. But one factor that is consistent is Joe. Joe and his unique perspective, his curiosity, and especially his tenacity that I want to talk to you about today. So what makes Joe a fantastic scientist is that he always look where no one else does, but still ask very relevant questions. Here is Joe looking at a solar eclipse through a pinhole. So to go from, to really understand what was in the nucleus of those oocytes and the nuclear body, Joe was interested in looking at the RNA in the nuclear plasm. So as he mentioned, he developed a technique to isolate nuclear plasm and also cytoplasmic fraction. And with a simple RNA-seq experiment of those two fractions, we found some really interesting data. I'm showing you here the RNA-seq experiments with gene coverage of a gene LORP4. We have on top the cytoplasmic fraction, the nuclear fraction, and then we're going to compare it with whole oocytes. As expected, in the cytoplasm, most reads map to exonic regions. You can see here in orange. And in the nucleus, most abundant RNAs were non-nuclear RNA, such as the small nucleolar RNA, snRNA. What was very exciting is to look at all those reads that map to intronic regions. Those, turns out, after much work, turned out to be stable intronic sequences. And we also were excited to find that some of those reads were found in the cytoplasm. And because we had the most perfect model system, we knew for sure that we were looking at stable intronic sequences. So one of my favorite moments around this time is when we were trying to come up with a name, that ends up now being stable intronic sequences or cisRNA. Joe thought perhaps we should name the cisRNA that were in the cytoplasm a different way. And I think he was really onto something, and we should perhaps have stick to it. He thought that those cisRNA should be called stable exported intronic RNA, or, you know, sexy RNA. Joe has the best words, actually the best words, but he also has the best curiosity, which is quite contagious. Here is Joe showing us in the lab a capillary that he got from his garden. Joe teaching students that were there for their science day. And here is Joe with Svetlana Deresheva looking, fishing some eggs from frog eggs. And the reason I say that Joe curiosity is contagious is because he gets us excited about any unexpected observation. One thing that we wanted to test is if whether cytoplasmic cisRNA were found in other species. As Joe would do, we've looked at the perfect model system, which turns out to be red blood cells from mammalian system. Red blood cells are enucleated cells, and therefore are a pure bag of cytoplasm. We did RNA-seq on red blood cells, and what was very surprising is that a third of the reads map to 7-SL-RNA. The reason it is quite surprising is 7-SL-RNA is a non-coding RNA involved in translation. And we know that red blood cells do not translate any mRNA. So Joe always encouraged us to use the right technique to confirm our initial observation. So here's a northern blot, and indeed we could find the full-length non-coding RNA, 7-SL. And this was definitely the perfect technique to look at, because now we know that 7-SL also can be further processed into a distinct fragment of 68 nucleotides, only in red blood cells of mammalian. Northern blot has been quite useful to find even more unexpected science. Here's a northern blot looking at SNO-RNA. It's a probe for SNO-RNA D69. And so we could detect the mature SNO-RNA as expected. But in the oocyte, we could also find a stable lariat that bear the SNO-RNA, suggesting that some of the lariats are not processed in the oocytes. And more excitement is coming up very soon on this, so keep updated. And one final trait that I want to talk about among the many is Joe's tenacity. And we've been thinking quite a bit on how to represent this in the picture. So we actually had to find a still frame from Carnegie's security camera to find Joe playing Zera on ping pong and winning, I should add. Joe always hits back a bit harder and with a spin. Joe is very careful in his experiments and tenacious, even if that means wearing gloves for an experiment while using Trizol or doing very tricky experiment. One example I will show you is showing that Joe always liked to use the newest technology. So when he's not developing his own technique, he uses the latest. And this is single molecule fish. He applied a single molecule fish after a lot of fails and tried. He got beautiful staining of intronic sequences in the cytoplasm of human cells, chicken, mouse, showing that sexy RNA is everywhere. And more recently, Joe is keep heading at this hard puzzle that never left his mind since his graduate work to understand the fundamental organization of our inheritary material. So this is a work led by Melissa Kenneth that with Joe are now mirroring cytology with genomics. Here's one of their latest preparation of a Lampbrush chromosome from the oxalotl. Joe, thank you very much for all the training you've given us and all the fun experiments that we've done, all the good memories. I wish you all the best. We all do. I want to thank the RNomics team, especially Eugene, who started this new era in the Gall lab, as well as all the people that helped me prepare this talk. Joe, we wish you all the best. Thank you very much.
Thank you, Gaëlle. Our next speaker is Alan Spradling, who will provide the closing remarks for this part of the program. Alan first met Joe when he was a graduate student at MIT, where he did, no dog, sorry guys, where he did his PhD with one of Joe's former graduate students, Mary Lou Pardue. Thus, although Alan did not have the good fortune of being one of Joe's former students or postdocs, he is a scientific grandson of Joe's. Alan came to the Carnegie as a staff member in 1980 and later served as its director for 20 years. He is also a Howard Hughes investigator. Alan's research focuses on oocyte development in both flies and mouse. Among his many honors, Alan is a member of the National Academy of Sciences and recipient of the Genetic Society of America Medal and the C.F. Conklin Award from the Society of Developmental Biology. Alan.
Thanks very much, Jenny. I'm not going to use a PowerPoint, so I'm very pleased and honored to be trying to do the impossible and summarize all that we've heard today. I would also like to stand for the many, many scientific grandsons of Joe Gall who have also been tremendously influenced by the things that you've heard about today. It's really been satisfying to hear Joe recount his career and illuminate with characteristic modesty and humor some of the principles of philosophy that has guided him so unerringly. We have listened with equal fascination to reminiscences and anecdotes from many of you who've played key roles in Joe's stellar scientific accomplishments. We've heard repeatedly about the joy of doing science within the Gall universe. It was fun just working together in the lab, and we also have seen today that your camaraderie is still going strong, despite the passing of years and lab generations. Training with Joe is not just exciting, but also a source of a uniquely valuable scientific worldview that has allowed so many Joe Gall graduates to pursue wonderful careers of their own, like newly forming galaxies, shooting in all directions after the Big Bang. We were further privileged to become better acquainted with Joe's childhood experiences that shaped his scientific and personal style. He found that one learns best by doing, and that you should trust what you see with your own eyes even more than what you read in the observations of others. Joan told us a little bit about Joe's interest in astronomy, as Gaëlle mentioned in one of her pictures. We'll hear later from Larry that this began early on in his life when Joe ground his own telescope and peered at the wonders of the night sky. Joe was flabbergasted when it was discovered that the main Hubble Space Telescope mirror, with all its scientific sophistication, had a defective curvature. Anyone who ever ground a lens could have detected that problem, he explained. However, Joe had chosen to study biology rather than astronomy, so he was not available to prevent that particular near disaster. But Joe has nonetheless taken on a unique astronomical role within the field of biological science. For in the universe of biology, Joe is the North Star. The scientific heavens may turn and swing with the fashion of changing seasons. But those of us who know Joe and understand the things he stands for in science hold steady and are always there to inspire and guide us. Are gravitation-like forces suggesting that groups need to congeal together into large masses in order to compete? Large groups and projects have made wonderful contributions to the advancement of our field. None of us would want to live without genome sequences, stock centers, and soon single-cell RNA-seq of every tissue and developmental stage. But Joe's work and voice reminds us how simplicity can be even more powerful and original. Joe asked if you can hybridize probes to DNA on a millipore filter, why not DNA on a microscope slide? If an oocyte nucleus contains thousands of nucleoli, maybe nucleolar genes are amplified. If some introns are preserved and moved into the cytoplasm, maybe they're doing something interesting. We need simplicity and clarity, because if all we have is stellar data, there may be no one around who can interpret it productively. Data are a given that will always get bigger and better, but scientists cannot wait and hope the data quality will eventually improve until some meaning becomes self-evident. Science is about developing novel ideas needed to understand our world, something that data only enable. It's often been said, oh, you couldn't do anything with the technology back then. But Joe never seemed to notice such limitations. He showed that no matter where a field's development stands, there are simple, revealing experiments to be done based on what is known and available. Yes, you may have to change the question you want to address, but there are always interesting, unexpected, and important results waiting for someone who knows the right answerable question to ask. For example, it's great to hear about the excitement these days about unconventional model organisms, but also a little amusing. Yes, it's easier to work with these systems now using genomic techniques, but for his entire scientific life, Joe has done important work mostly outside the specially favored model systems, such as Drosophila melanogaster, where he started his career. Joe wanted to make use of each organism's special properties and was therefore drawn to newts with chromosomes more tractable than polytene chromosomes, Tetrahymena with telomere proliferation, the water beetle Dytiscus, whose oocytes had a giant DNA staining body, Drosophila virilis with disappearing polytene heterochromatin, Sierra caprophila and Rhynchosierra with DNA puffs, as well as xenopus, salamanders, paramecium, euploides, mice, snails, coelacanths, gastrothecae, and even human cells, and that's just among the animal kingdom. Throughout his career, Joe has made research on unconventional creatures seem simple and normal. He showed us that whatever the current level of technology, by choosing the right organism and the right question, one can uncover a new aspect of nature. Ultimately, Joe and his career also guide us beyond just the subject matter and questions of scientific concern. The way you organize your scientific activities can be as important as the subjects you study and the discoveries you make. Very few of us are able to add anything to the existing body of knowledge on our own. Doing science, like every human enterprise, is about the people you work with in our laboratories, in our institutions, and in our disciplinary communities around the world. Working with others adds immeasurably to the joy of science, but also raises the question of who is selected to be part of our ventures and how the rewards of success are shared among our group members. Our understanding of such social organizations seems to have much in common with scientific knowledge itself. We always feel we have a better grasp of what is fair than in previous eras, but over time we realize much still remains to be improved. Joe has always run his laboratory in a humane manner far ahead of his time, in a way that benefits both science and all the participants of the enterprise. Scientists need and deserve to take pleasure and pride in what they've accomplished. Knowledge is not acquired easily or without many disappointments along the way. Joe shows us how to enjoy the thrill of discovery while remaining humble and recognizing the many contributions of others who worked on the same questions. In closing, Joe, we thank you again for making us better scientists, better human beings, and for all the good times we've shared together. This represents the conclusion of the formal talks. However, after a 5-minute break, please everyone return for a session of open discussion where we hope to hear from many more of you. Thank you.
Do we think we should do the 5-minute break, considering we've overrun?
Sarah, give people 5 minutes and then we can start. And then since we're running late, keep your eye on the clock. So shortly after 2 o'clock our time, or if you want to go a little later, that's only 18 minutes from now. You should conclude that and ask people to send you or to you, Ginger, or the website, their anecdotes if we don't hear all of them.
I think what I'll do is just give a quick tutorial of the website. So I'll just share my desktop and just show that, and then turn it over to the family.
Yeah.
Yeah.
And then a few people from the audience, and then we'll call it a day.
Great.
Sounds good. I'm taking a bathroom break. I'll be right back.
Okay. So Naveed, are you currently online?
Hey, Sarah, did you have a question?
Yeah, so what I'm sharing right now, are you seeing the
I'm
website?
seeing the website, yep.
And nothing more, not on any of the participant panel things that I have open and all that?
No.
Great, great, great. And can you see my cursor if I'm on the website and what I'm doing on it?
I can.
Oh, perfect, perfect. Okay, thanks. And so people will raise their hands and I'll be able to just yield to them, or you're gonna just follow that sequence anyway with the family members.
Talking to me?
Yeah.
Yeah, after Joe's family speaks, then whoever wants to raise their hand from the audience, you can promote them to a panelist, and then they can turn their camera and mic on.
Right. So to confirm, it'll be Larry, who will yield to Jen, who will yield to Barb, who will yield to Shelby, who will yield to Diane.
I don't have the list in front of me, but I can check.
Okay. Oh, so that would be the order.
Okay.
Okay. And I can if they raise their hands, I can enable them.
Yeah.
Or
So
I can
if
pick
they
them from a panelist and promote them.
exactly, and if they raise their hands, it'll pop them to the top of the list, and so it'll be easier to find them.
And even if they don't raise their hands, I can just find them and then.
Yeah, we can scroll down and look.
So it seems to be like if I hover over their name in the attendees thing, it just gives me the option to give them to chat with them.
All right, here, let me make you a co-host, and then it'll give you the.
Thanks.
Yep. There you go. You should be able to do it now.
Yep. Perfect. Perfect.
Sorry about that.
No problems.
Zara, this is Larry. I think we have confirmation on all the speakers from the family too.
Perfect. Looking forward to it.
Zara, can you hear me?
Yes.
Ginger, just remember, I am introducing you.
Ah, yes.
Okay.
So then I'll turn off my screen. Let me do that. I'll stop my screen share and I will yield to you then, Ginger.
Zara, 5 minutes is up, so if you want to start, it's fine.
Okay, so I'll let Ginger start up.
We hope everybody had a good break. I'm now going to introduce Zara Nizami, who will lead the open mic session. She is also a co-organizer of this meeting, and she's originally from Hong Kong. After graduating from Princeton in 2005, she became a grad student in Joe's lab, where she worked on nuclear bodies in Drosophila and Xenopus. Among her PhD achievements was her co-discovery of cis-RNAs, stable intronic RNAs, or sexy RNAs, that you've already heard about from several speakers. After her PhD, she stayed in Joe's lab as a postdoc, switching to the study of gene expression in amphibian germinal vesicles using genomics and super resolution microscopy. In 2012, she was awarded the Johns Hopkins Service to Science Award in recognition of her science outreach. In 2016, she won the Carnegie Postdoctoral Invitation and Excellence Award, abbreviated PI. Apparently this award ceremony involves eating a lot of pie, at least from the pictures I've seen. The PI Award recognizes achievement in science, education, and service. Zara is currently an applications development scientist at a British biotech company called Oxford Nanoimaging.
Thanks, Ginger, for that intro. I really appreciate it. So this is now the open discussion section of the symposium. And I know we've all been eagerly waiting for it. And I apologize that I think we won't necessarily have time for all of your insightful comments and anecdotes that are going to come in. But we have prepared for this eventuality. And what I'm going to do is I'm going to quickly share my screen so you can see that we have a website where you are invited and most welcome to upload videos, photos, anything that you have. I hope you can all see this right now, but basically if you once you get to the website, which is hosted by Carnegie, this email link would have been in your original email blast, and it was also on the poster. If you don't have it, feel free to email one of the organizers, we'll send it to you. Then you can tab over to photo gallery or to the anecdotes section, and you'll see that there's the buttons for you to upload your information. And if you were to do that, it actually, it'll go to a Google Drive account where you can just dump everything here. You can either do it in an organized way or, you know, whatever you want to do, just dump it here and I'll take care of it later. And in the coming weeks, I'll collate it all and then I'll send out another email blast telling everybody, oh, it's ready. Everyone come and check out all the anecdotes. And we can all systematically enjoy all of our thoughts and all of our praise and all of our thanks to Joe for his amazing contribution to our careers and to our lives. And speaking of lives, as you all probably experienced, as I did, Joe really treats you as one of the family, and his family treats you as one of the family. And we all love this portion of the symposium when we transition over to the family to discuss, to talk about Joe and life with Joe as a scientist and so forth. And so I'm very excited to now begin this part of the program where we will be hearing from Larry Gall and Jen, his daughter, and then followed by Barb, Joe's daughter, and Barb's daughter, Shelby, and then Diane. So without further ado, over to you, Larry.
Thanks, Zara. Can you enable my screen sharing? Thank you. Hello, everyone. Can you see Joe's initials here?
Yep.
Well, it's wonderful. Thank you, Zara, for that introduction. Good afternoon. It's a real pleasure to be here today and to join this fabulous and enthusiastic celebration of my dad's career with several generations of his extended academic family present. On the call are also several generations of Joe's literal family, so we will offer a few additional reflections. Now, I have to say that you've got this all wrong, all of you. Retirement is not a concept I would use in the same sentence as Joe Gall, or Diane for that matter. And I can't imagine that Joe has any intention of retiring. He will simply continue to do absolutely everything of interest to him, just in nominally different modes. Now, you all may think of Joe as an embryologist and a cell biologist, but as you probably all can appreciate from Joe's looking glass talk and the testimonials from the earlier speakers today, for my entire life, I've known him as a naturalist and a historian of science, with an abiding interest in whole organisms, what they do, where they live, etc. And this has informed his thinking and approach to science from the very beginning, as many people have been mentioning. Here you can see this manifested when Joe was still a relatively wee lad, always on the lookout for new things. Of course, everyone grows up. As he grew up, the focus broadened into other disciplines, such as astronomy and optics, as is the natural progression of things. And as Alan had mentioned, I believe this is the telescope that Joe and his brothers ground together. And of course, those interests usually end up sticking with you throughout adult life, pondering the universe. And Joe transferred from that one telescope to this Questar, which he still has to this day. And as one goes along, one gains wisdom, but not necessarily fashion insight, I would have to say. But returning to natural history, that's the bond that my sister and I have always shared with my dad from our early years. Nowadays, tragically, most children have less exposure to the outdoors compared to their parents, and certainly their grandparents. But thanks to Joe, it was okay and encouraged to run feral all day through the fields and woods and haul critters back to the screen porches in the houses where my sister and I grew up. This is 142 Arthur Place, where we lived when Joe was at the University of Minnesota. This experience ended up shaping both of our careers, Barb's as a veterinarian and mine as a museum curator. And as such, I'm constantly on the lookout for acquiring and preserving additional collections. And I rue the fact to this day that the corpus of my dad's childhood insect collection are no longer around. However, at Yale, we do have enticing tidbits from that time frame. This is the earliest surviving insect specimen that Joe collected. It's dated 1941. It's an Isle moth from the family home in Upperville, Virginia. The cool thing is that this is a mosaic gynoendomorph, as you can probably tell. It's a mix of male and female characteristics. And these are extremely rarely captured in the wild, as opposed to rising in lab broods, and so on and so forth. During his time in New Haven, my dad donated a smattering of other insects to the Yale collections, mostly butterflies and moths, that he had collected while traveling to conferences around the country and the world. This would include 5 continents, 5 countries, and at least 6 United States. Alongside his students, colleagues, and publications, these specimens own a place as part of Joe's permanent scientific legacy. And in closing then, the natural history theme continues to run strong from Joe all the way into the F2 generation of his family, shown here together at Barb's home in Westminster, Maryland. There are the F1s. Here are the F2s. So Dad, congratulations on your well-deserved non-retirement. I expect little to change, and I look forward to your next text message about Lepidoptera from your backyard. And to Joe's many students over the years who were and are still very much a part of the family, thank you all for wonderful memories. I'd now like to pass the baton to one of Joe's F2s, my daughter, Jennifer.
Awesome, thanks, Dad. Hi, I'm Jen Barrigal, the eldest of Joe's 3 grandchildren, and I'm very happy to be here in celebration of Joe's career. So, as you can tell in the next slide, my artistic understanding of the genesis of an axolotl differs quite a bit from Joe's interpretation. And I'm sure we could all tell a myriad of stories about Joe, but in situations like this, I'm here to add the personal and I hope comedic touch. Obviously, I have a lot in common with my dad and grandpa, a love of puns, clearly, crossword puzzles and limericks. But as you can see from the photos, I twirled in a different direction. Though I've always had a real appreciation for the natural world and enjoyed all sorts of outdoor activities, science in school was never my favorite. I still have trouble telling mitosis from meiosis from my own neurosis. I was the student who was happy to give the reins to my lab partner to do the experimenting, dissecting, or whatever it was that we were doing. And I'd meticulously record information and, of course, write up the entire lab report. Essays I loved. Science classes, I had awesome teachers, but the content wasn't always of interest. I was a professional ballet dancer for several years, and when I went back to school, not surprisingly, I pursued my love of the humanities. The focus of my undergraduate research was on 16th and 17th century European travel and trade, and that's how, interestingly enough, I discovered a real passion for science. Because the era of exploration and exploitation coincided with and influenced the move away from medieval scholasticism and the rise of inductive reasoning in Europe's academic circles. I found myself fascinated by the development of what we call modern science, tracing the history of natural philosophy from ancient Greece to Islam's golden age, and then to the European scientific revolution, which, by the way, depended on individual thinkers' curiosity and genius, but also the intersecting forces of empire, colonialism, and religious upheaval. Joe and I had great fun at his 85th birthday symposium, recognizing that the very people whose work I studied were authors of many of the rare manuscripts and books Joe has in his own collection. We could talk about the drawings of Maria Sibylla Merian and the oddities of Leeuwenhoek's studies, or the vast collections of Hans Sloane, or the cultural significance of Darwin's theories. And so now, as a high school history teacher, one of my favorite topics to teach is the European scientific revolution. And earlier this spring, I had the privilege of welcoming Joe and my dad to a virtual live Q&A with my 9th grade students, where we began our discussion with 3 very simple questions. Who are you? What do you do? Why do you do it? My students loved it. We had an incredibly engaging conversation, and I'm thrilled Joe could share his love of microscopy and biology with my students. I think they have learned at a much younger age than I that learning is interdisciplinary, and that the history of science is pretty darn cool. So Joe, many congratulations on an impressive career, but more importantly, thank you. It's clear from the many people present here on the panel and all those watching from home across the globe that you are an influential mentor and teacher in both your professional capacity and as a grandpa. Thank you.
Hello, Barb.
Thank you, Zara. Thank you, everybody who's organized all this. Zara said, you know, Dad's family considers you family. We've always considered everybody on this Zoom call and everybody around the world family. Some of my earliest memories are from dinner parties, and I would be a little girl and I'd be serving appetizers or something around the world with, I didn't know it then, but soon to be world-renowned scientists. And just, you know, I remember absorbing all I used to sit quietly in the background and absorb everything that was being said. It's one of my best memories, and just love having all of you out to our property in Westminster. Everybody who's visited, everybody's always welcome. I don't have photos right now, but I did pick out lots of photos, just didn't get a PowerPoint done. I will upload the photos. I'm glad to have the opportunity to do that. But I'll just piggyback on Larry's sentiment. When I first heard that Dad was retiring about a month or so ago, we were shocked. And I thought, this can't be, this will never happen. Little disbelief, little worried. What does this look like? Thinking, you know, this is a person, my father, who's always said he never went to work a day in his life. So, and I like to keep that mantra. I think everybody, if it's possible in their life and their career, should be able to say that they've never gone to work a day in their life. They've just loved what they do every day. So what was he going to do? So there's always the insect world outside to inspect, correspondence, organizing a life of discoverings and firsts. He's always just so genuinely fascinated and passionate about the scientific world and discoveries he was making that every day I think has filled with wonder in his life. And I think, you know, that definitely was passed on to me, and I know Larry and my children, and hopefully my children's children, to be just passionate about nature and the scientific world. I've always thought of Dad as kind of a Renaissance man, rarity in the 21st century, I think. Definition being well-rounded, versed in many areas, someone whose goal is to reach their highest potential in life and be well-versed in many different subjects. Given that he could, when I was in high school, and probably still can, help with the Latin declination, I think this title fits. So, you know, I don't know anybody quite like him, as I'm sure everybody would agree. So typically one thinks of retirement is you come home from the office one day, you take a cardboard box, you fill it up with your stuff, family photos, a few tchotchkes, pens and pencils. In Dad's case, I just had a vision of him coming home with this collection of frogs and a carnivorous plant that used to be on his desk, his Venus flytrap. So just a little different vision. But we were immediately relieved to hear that retirement was meant downsizing, not leaving the laboratory environment, because what a relief. It just wouldn't seem fair or right to deny him access to a lab and a microscope and continue to correspond with his colleagues all around the world. So growing up, Dad always took time to participate in my interests and those of my family, looking at lots of pictures over the last few days. And most of the pictures, Dad is in some kind of teaching mode. He's in his glory collecting muck from our pond. There's many pictures of a net going into our farm pond with bunches of muck and being poured through, and specimens picked out to take to the lab just to look at them, just to look at their DNA, just for the fun of it. And my children participated in that as well. So the wonder of nature is being passed on. So he's fostered my daughter's love of learning. He even got on a horse every now and then for me, although his appreciation of my sport was mainly from afar. So I've not been directly involved in the lab, but the influence of the lab and the science was always felt in the household, and always a standard was set. I live and work in more of a macro world of veterinary medicine, but I have strong roots in the microscopic world of the cells and genes that are responsible for the diseases that I treat. So I have a little bit different perspective than some of my colleagues sometimes. I'm definitely known as the nerd. So as Alan said earlier, I got to hear some of his clip that Dad learns best by doing, and I think I've definitely inherited that gene. That is the way I learn, is doing, exploring, analyzing, but not just reading. I want to say just in, kind of conclusion, there's this huge gene pool of scientists who are mentored and encouraged. I didn't get to hear everybody today, but I'll go back and listen. Had their careers and their scientific discoveries just launched from the Gall lab, Minnesota to Yale, Carnegie, around the world. So with retirement, I'm sure this doesn't mean that my dad's not still going to be influenced, but it means it's up to this elite and quite large and diverse group to carry on in the same vein. And I've always seen in the lab open sharing of knowledge, professional camaraderie, holding up a standard of excellence for the scientific community, and which I think is rare. I think I've been in enough research environments to know that that's not something that doesn't happen unless it's intentional. There's also this large gene pool of family and friends who have been and continue to be mentored and encouraged in their endeavors. So thank you, Dad. Thank you, this whole community. Retirement means just something different. It doesn't mean stopping. I know that to be true, because it just wouldn't be fair to keep you away from like I said, the lab and the microscope. So thank you again, everybody who organized this, and I'm glad I was able to hop in, and I look forward to hearing some of the comments.
Thanks, Barb, that was great. I'm trying to look for Shelby on the attendees list. Is she on there? Is she going to say some words as well? In the meantime, maybe I'll promote Diane to panelist to share some words. I don't know if now, Diane, you notice that you have supreme power.
Can you see me?
Yeah.
Okay, good. So I just wanted to jump in here and say hello. I'll be very brief. It's really been my honor to travel together with Joe over these years. And for the last 5 months, we've been locked down together. So I think that you all would agree with me that there are few people that you'd rather be locked down with for 5 months than Joe Gall. And so it's been a real trip for us, as moving some of his lab things as well. But mostly I wanted to just say that you all have been our extended families over the years, both professionally and personally. And we've had a great time being with you for parties, both at our home, at your home, at the lab parties, visits to the woods, and both on national and international travel. just been wonderful to join with fun experiences. And I guess I wanted to say that the one theme that I've used is change, and that there are two things really, just that some things change and some things don't change. And the things that change in Joe's world and life are basically the colleagues that move through the projects, certainly the technology that's evolved over the years. But then what stayed the same, I feel, are the personal relationships and the caring that you all have for Joe and that we have for each other and that we have for you. So I think that really, in addition to what Adrian said about the scientific and the fun and the excitement of learning, I would add both the love, respect, and patience that I believe is the hallmark of Joe's approach and that of his whole extended group. So in that, I'll close, and I just want to add my thanks to Joe, who's sitting next to me, and also to all of you as our extended family, and thank you for this event. Sarah?
Thanks, Diane. Thanks for that. And I'm going to now promote Lily, Barb's daughter, to panelists, if you want to say a few words. I know Shelby had wanted to say something, but she had to pop out.
Yeah, I guess I can say a couple things. I didn't have anything prepared, but let me clear my space real quick. Okay. Hi, everybody. My name is Lily Idell. I'm Barb's oldest daughter and the middle grandchild. Didn't really have anything prepared, so we're going to make this quick. But it truly has been such an honor to hear all of your comments and collaborations with my grandfather, because as the family, at least the third generation down or second generation down, we weren't as involved in the actual lab stuff. We would see his lab, but it's really cool to see that different perspective and just the respect and how much his grandpa's personality and his kindness and his loving and his caring nature towards everybody really reflected in the workplace as well. And we're able to see, the lab family really was our family. We had multiple Christmases over the last probably decade where we would just invite people from the lab to come and have Christmas dinner with us. And they would bring something non-traditional, but that was the beauty of it, is like we were able to just spread the love throughout the different cultures and the different just different lifestyles and people. We're not all scientists. But yeah, kind of what my mom said too, we just thank you for being here and saying kind words, and definitely have had an influence on me personally. I did not go the science route in my life, but it did inspire a love. My grandfather did inspire a love for nature as well as my parents, to be outside and ride horses and just to go exploring in the mud and the muck and catching frogs in our farm pond at all wee hours of the day. So Grandpa, happy retirement. Sorry, it wasn't a whole lot here, but yeah, I know we'll still see you and hang out, but just thank you everybody for being here and speaking.
Thanks so much, Lily, for the impromptu words that were very moving, and it was really nice to hear you speak about that, about your grandfather with so much love. So at this point, we've run over. That's perhaps not surprising, but I think for all of those virtual participants who are hanging on, we have some hands being raised. I'm going to call on, let's see what we got. Feel free to use the hand raising tool. Oh, they're coming up. Okay, well now where do I, oh, there they are. Okay, Julio, I'm going to promote you. Here you go.
Hi, can you guys see me?
Yeah, we can see and hear you.
Okay, let me just share the screen. I just wanted to say thank you to Dr. Gall. Let me see if I can share his screen. It is 3:20 a.m. in Japan right now, where I'm currently at. So yeah, I was a rotation student in Dr. Gall's lab in 2006, 2005, I can't remember exactly, but and I was at Carnegie until 2013, I believe. But all throughout the years, I interacted with Dr. Gall. It was a pleasure, you know, taking this microscope course and a lot of little other stories, little anecdotes, such as the transit of Venus, that he set up his little microscope outside the Carnegie. And even, I mean, I'm really tired right now and I don't really know what to say, but other than thank you, arigatou gozaimasu, Dr. Gall.
Thanks, Julio. I'm gonna downgrade you now.
Okay.
And Katie Mitchell, you're up. Can you hear me? Have you managed?
Hello?
Yeah, we can hear you. Go for it, Katie.
Awesome. There's video. Hi. So I was in a lab, 10-ish years ago. And I just wanted to bring a guest who was also made an appearance in the lab that Joe and Zara met. This is Mocha the ball python. She is 27, so she's aging basically about the same rate that Joe is. And just thank you. I always loved all the critters in the lab, and everything was, you know, super fun. So I just thought you might like to see the snake, because I remember when I first brought her in, your eyes lit up and you leaned forward, you know, to like try to be like, oh, I'll take her to hold. And I just thought that was really sweet, and I appreciated how much you loved my snake. So that's it.
Classic words, Katie. Thank you for that. And I'm going to, and also Snake's looking great. All right, I'm promoting you, Marnie. You're up next. Thank you. Hi, Marnie. Can you hear me?
Hi. Can everyone see me? Oops. Yes. Can everyone
we can
see
see you.
me?
Yep.
Okay. Thank you. It's a real honor to be able to speak today. First of all, I really want to thank the speakers, especially those incredible women who started this symposium off. It's not just Joe who's been a role model for me, but it's really his amazing female trainees who have inspired me so much. I think that's what's so incredible is the legacy that Joe has given us all, other than just the direct training. I did want to just add two small things. First of all, I've learned so much from Joe, and one point that I don't think anyone else touched on is when Joe asks you those embarrassing science questions that you should know the answer to, but you haven't really thought about it until Joe has pointed it out to you. And this happened to me at the Carnegie once, where I had some transgenic fish that had a histone labeled, and Joe said to me, well, have you looked at those fish to see if they label chromosomes and oocytes? And I looked at the literature and I thought about it, and I realized nobody had ever really looked at this transgenic to see if you could see labeled chromosomes with GFP in oocytes. So I had the pleasure of dissecting some zebrafish and actually looking under the fluorescence microscope and learning what I probably should have already known, except I needed Joe to point it out to me. It's been really fun to send Joe and Diane pictures of the various amphibia that we've come across in New Hampshire. And I'm proud to say that I now too have seen a red-spotted newt, or the eft stage, which I recently learned about from Joe. And finally, I just want to add, outside of the lab and all the great science stories and so much I've learned from Joe. I first met him when I was interviewing for graduate school at Yale and was so disappointed to learn he'd left Yale just as I arrived to go to the Carnegie. So I had to follow him to the Carnegie. But I just want to add finally that Joe and Diane are just such great friends, and it's just wonderful to spend time with them, whether we're exploring dinosaur museums in Canada or going on nature walks. I just feel so privileged to know both of you. So thank you very much for this great symposium, and thank you, Joe, for enriching everyone's lives.
Thanks, Marnie. That was great. And I think I'm going to call on Harry next, promoting you to panelist. Can you hear us?
Okay, can you hear me?
Yes, we can.
Great. Thanks for letting me speak today. I appreciate this opportunity. You know, when Susan was reaching out for anecdotes about Joe, the one I recounted to her was when Joe ate DNA for a public forum in 1976, and she had never heard of this and asked me to get some proof of this, and I couldn't find any. All I could find was an article in the Harvard Crimson about how Cambridge had shut down any activity on building a P3 lab, but Joe and Frank Ruddle, then the chair of the department, were interviewed about how things were going in New Haven. They said they weren't going to have any problem. And what I remember hearing in 1976 is he went to this public hearing with New Haven government and Yale, the Biologic Safety Committee, and he precipitated DNA and ate it. Now, I couldn't find any documentation of that. And I really searched. I Googled everything. I used different search engines, different word combinations. And I started to think maybe this was suppressed. I started thinking maybe it's the Mandela effect that I've recently learned about, where people remember something a certain way, but it never actually occurred. But I'm really pretty sure it happened. And in fact, I'm pretty sure it must have been Xenopus, the Xenopus clawed toad DNA, because people in New Haven remarked that after that public forum, they never again saw Joe Gall without something covering his feet. So it may have been Xenopus DNA that he actually ate then. You know, Joe, what I want to share with everyone is what your legacy means to me specifically. And that is, I never was a grad student or postdoc in your lab. I was a research assistant. And I started in 1974 as a high school student working with Kathy Carr, and left in 1981 to go to Stanford for my MD PhD training. And I often wondered how that happened. I didn't come from a science family. My dad was a truck driver. He was a compulsive gambler, and I later learned in life he was actually an ex-con. My mom was a bookkeeper, but she had something in common with your mom, Joe. She really inspired all of her children to get an education, to pursue their intellectual development, and like your mom, made it possible for me to get a microscope, even though her salary wasn't one that would really allow her to do that. So I think maybe that had something to do with you selecting me to be in your lab working as a senior high school student with Kathy Carr. Of course, the other theory is that everyone knew I was chasing your daughter Barbie. I've not made that a secret. Nowadays, it would go by other pejorative terms such as stalking and creeping, but it was truly quite pure, and it was just chasing your daughter. I don't think it was that, because you knew, according to, you knew the lyrics of the Tom Petty song that protected your daughter from me, and that was the Tom Petty lyric, "She likes horses." And Barbie, as you could tell from that last picture, loved horses, and that was her main love. So I moved on into the lab. When I got into Yale, he knew that I came from a rather poor family, and offered me work study in his lab, where I did a whole bunch of different things. He allowed me to be a teaching assistant at the Cold Spring Harbor courses of molecular cytogenetics with Mary Lou Pardue, where I got to teach what I had learned from his mentorship. And then 1981, looking to apply everything that I had learned through him and all of his grad students and postdocs, I went to an MD PhD program trying to apply that science to human disease. And I worked with Larry Kettis there, where I got my PhD looking at human actin genes, but ultimately chose clinical medicine. And Joe, here's where my final comments come for you, because I remember growing up in your lab that there was a little bit of a, let me say, dislike or for MD scientists. And I'll tell you, I often felt like, did I sell out? And I want you to know I didn't. I really do. I want you to know how much my time with you meant, because now I'm one of the few attendings who could sit at a microscope and know how to focus a condenser and adjust a diaphragm so I can look at leukemic cells and blood cells with residents and fellows. I take every opportunity when we're talking about a FISH analysis to identify a certain cytogenetic change that makes patients more sensitive, their disease more sensitive to the addition of an antibody to their chemotherapy regimen. I take that opportunity to tell them I had the privilege of working with Joe Gall and Mary Lou Pardue, who developed that. And it's important that I had this background because I now have the background so that I can talk to the scientists who are developing the novel targeted therapies that are helping so many people with leukemia. And so for the very small part that I've played in the development of novel therapies, it really is you. My last two comments: when I was a high school student, I didn't understand that there was a difference between women and men in terms of academia. I saw Joe's lab. Everyone was equal. And what they had in common was a passion for pursuing scientific excellence and investigation. It had nothing to do with gender. And so in my position now as the chair of an NCI-sponsored leukemia committee, I have the ability to promote women into those positions of clinical investigation, not because only they're women, because they're at the table and they show the passion for doing the work and the interest in doing the work. And so finally, what I'll say, Joe, is your legacy is going to go on in my life, in every patient I touch, because you help me think outside the box. And so for every patient that I see and whose life I'm able to extend for some amount of time, I never would have been here if you didn't give that high school kid a chance. And for that, I'm thankful. And what you've just heard is just one story. And what you're hearing from all the people today are all the people that you trained that then go on to do good in the world. Thank you, Joe, for everything you've done for me and for us. And I know you're not going to retire. Thank you.
Thank you, Harry. Yeah, I wonder what DNA tastes like. So that's great. Okay, so I'm going to call on Steve Farber to become, I'm going to promote you to panelist at this point. So you can hear from another colleague, and then we'll end with calling on Xin Chen, who will speak as a colleague and a collaborator. All right. Steve, can you hear me? Are you ready?
I can hear you. Can you see me? It says video. Can you? What'd you say? I'm seeing a black screen, but you all see me?
I see you.
You're good to go.
Okay. Well, we've heard so many stories about Joe's scientific accomplishments and his mentorship of folks in his lab and from family members, but I felt I'd add a view of as a colleague who has had the privilege of working in Carnegie with Joe. And a quality I would just like to bring up is he's like a wise sage on so many dimensions that are not specifically just about the science in the area of his expertise. And often that's so important to young scientists coming up, is how to navigate an academic career, how to handle difficult reviewers, how to mentor students when challenges come up. And my point is not a long one, but Joe, you've been always the person who, if I want to get an opinion on what to do, I walk up to your office, and usually you'll drop everything. Occasionally you'll say, "Can you come back, Steve?" But most of the time, and when you listen with all your heart, and then you provide me such valuable insights on all of these like pieces of a puzzle to be successful in science, and how to manage a lab and how to manage challenging situations. And for that, Joe, that's something that I only hope to be able to provide to others, some of those skills that you have taught me. So just a super amazing thanks for all that you've done to teach me how to be a PI and how to be a good scientist.
Thanks, Steve. Appreciate that. I'm going to upgrade you, Xin, now to panelist. Can you let us know if you can share your screen and audio?
Okay. I think people can hear me, right?
Can hear you, yep, and see you.
Yeah, great. Yeah, good to see you there. And it's really a great pleasure to listen to all these amazing stories. I'm really speaking from like an academic neighborhood from the Carnegie Institute. So in the past few years, we also have this privilege to collaborate with Joe's lab. So Joe is really the driving force for this recent collaboration between our labs. Before working with Joe, we had a lot of difficulty in visualizing. We want to ask how histones are incorporated during DNA replication, and we were really ambitious trying to use some other microscopy methods to visualize this. However, Joe is so keen and so insightful to point to me, Shane, I don't think you need all these amazing, fancy tools. I think the STAT microscope we just got at Carnegie, you should give you sufficient resolution to give you a clear image. So really, he's absolutely right. So thank Joe and thank Carnegie for the generosity. So very soon after that, a student from my lab, Matthew Wooten, teams actually with Zara. It's good, great team, and together they have got really amazing results. And this collaboration leads to a recent research paper and also a method paper that is published just this year. And this method also now become a standard tool for us to study chromatin biology using cells from tissue, not from petri dish. And also, we just cannot thank Joe enough for this, his enthusiasm and inspiration. So every time we had a meeting with Joe, his eyes just widely open, and he's fully curious about all kinds of results, especially for odd results. So given all his insightful kind of feeling of the biology, this is really inspiring for us as a younger scientist to see. And I'm sure, I know Joe is also collaborating with my other colleagues in my department, and I'm really kind of speaking for all the colleagues from our department to the generosity of Joe. And also, for our graduate student program, Joe used to team teach with a group of us for a graduate core class. And actually, Joe, we have the privilege, he actually teaches microscopy to our students. And I often see Joe very often at all kinds of committee works, including thesis committees, GPO committees, whatever you name. And he's always there. Thank you, Joe, and happy retirement. But I'm sure, and I look forward to discussing with you more in the future. And yeah, that's it. And thank everyone for listening. And it's really a great symposium. And thank everyone for organizing and participating. Thank you.
Thanks, Shan, for your talk, for your remarks. Okay, so I think with that, I think I'm gonna conclude the open discussion portion of the symposium. At this point, I'd like to thank everyone for participating. I think I'm gonna pass it on to Susan Gerbi, who's gonna give us our concluding remarks to help us end the symposium. So over to you, Susan.
Thank you, Zara. Do I need to do share screen for full view, or you just do that?
I think if you could do that, that would help, because I'm not sure how I could do that for you. I'm sorry.
Yeah, but I don't have a PowerPoint.
Oh, you don't have it. Okay, let me see. I'll promote you to a panelist. I'll try to do. Okay, somebody's doing this for you. This is great.
So if you just call on me like you called on others.
Do you have a presentation to share, Susan?
No, I just wanted to say a few words, but I wanted to the screen.
You're good. You're there. You're full screen.
Oh, sorry. Yeah, you were there. Sorry.
Okay, I just see a black box. Okay, so as long as you can see me. So I wanted to mention that I regret we didn't have time for all the anecdotes, but they are being collated into a scrapbook. Zara already told you that you can go onto the Carnegie website on the Gall webinar to post your photos and anecdotes. If you have any problems with that, you can email the anecdotes to Ginger, who's collating them, and her email address is bzakian@princeton.edu. I wanted to close by thanking Yixian for having the idea that we should have this event that originated with her. I wanted to very much thank Navid for all the IT help and coordination of the event. I wanted to thank the committee of Ginger, Zara, and Zhilong who helped in organizing it. And I wanted to thank all of you for your participation in the event. And most of all, of course, I wanted to thank Joe, without whose inspiration that's influenced so many of our careers, we would not be celebrating this event. So as many of you know, we've had once every 10-year birthday symposia for Joe, starting with his 65th birthday at Yale, and then 75th and 85th at the Carnegie. And when he was approaching 90, I asked him, well, should we do one every 5 years and do the, have a birthday symposium at age 90? And Joe said, no, let's wait until I'm 95. And so I look forward to welcoming you all back for Joe's 95th birthday symposium. And in the meantime, Joe, we wish you lots of happiness in your non-retirement retirement. Thank you all and have a good day.